we'll get started. We've got an all-star and diverse group. The uniting theme is we're all affiliated with anesthesia departments. So I'm at Stanford. The next speaker will be Tony HUDETZ from Michigan, again in their anesthesia program. Then KATHLEEN is going to join us from Harvard's anesthesia MGH group. And then we've got another guy, Hami from Michigan. And we're going to end with our fearless leader, Stuart Hammeroff, will round out the end of the program. He was a late entry, but he asked if he could put a few words in for our workshop. And so that's what we're going to do. So I'm going to begin by telling you about what I think the most important effect of anesthetics at loss of consciousness is. And we've studied this in people and in animals, and I'm going to reduce it down to single cortical columns today. So very high resolution recordings, but maintaining a theme from humans to rodents. So without further ado, when I say brain slices, I mean we take our rat's brain and we make 500 micron thick slices and put them in a dish, keep them alive with all the nutrients and all the right things that neurons need, oxygen, etc. And they live and thrive for a long time. In fact, you can culture these slices and keep them around for weeks or months. But we do a daily type experiment, so just a few hours. So we put these slices in a dish and we're able to record individual neurons in high resolution. So that's seen in this slide. The blue trace at the very bottom is a single pyramidal cell that's generating a theta rhythm, and I'll tell you about how we do that. And the theta rhythm is a field potential generated by hundreds of neurons, okay? So we can show that simultaneously together with the individual neuron being recorded from. And as you can see, the theta rhythm in green is mainly being driven by the synaptic excitatory currents that are coming into the cell. So on every theta peak, the neuron has an EPSP. Every now and again, it will fire an action potential on the top of an EPSP. And you can see that has hardly any effect on the local field or micro EEG signal that we're recording. So individual neurons don't contribute a whole lot to the overall EEG field potentials we're recording. You need hundreds of neurons to generate the kind of amplitudes that we see in vivo. So at least hundreds of neurons. So how do we make these theta rhythms and such like? So we isolate the tissue into the brain slices, and then we can use high resolution patch clamp recording and visualization for placing electrodes where we want them. And we can stimulate electrically or chemically with neurotransmitters, for example. And so it's a isolated prep. There's no thalamus. There's no, well, very little thalamic input or input from other regions of brain, including other regions of cortex. Most of those connections have been severed. So it's really a little island of cortical tissue that's functioning on its own. So for example, we can generate the gamma oscillations that are associated with alertness and concentration and focus. and things like you see in an active brain. We do this by stimulating kainate receptors, which are a type of excitatory glutamate receptor that's prevalent throughout the brain, not just in cortex, but from spinal cord to cerebellum and on up. So these are very prevalent synapses. They're used to communicate fast excitatory information among nerve cells. So in this graph, you can see the raw signal at 40 hertz. Down below, you can see a spectrogram that shows you know, it varies over about 20 hertz, plus and minus 10, around 40 hertz. This is very similar to what gamma oscillations look like in vivo, including in people. Now, what about acetylcholine and the theta rhythm that I showed you just a minute ago? So if we apply acetylcholine, we produce a theta rhythm that you can see off on the left, starting at time 0. And there's two components to it. There's a slow theta and a fast theta. If we apply in the middle of the acetylcholine trial serotonin, or 5-hydroxytryptamine, it immediately shuts down the slow theta, leaving only the fast theta with a peak around 9 hertz or so. Now, why is this interesting? Well, that's exactly what happens in an awake, freely moving rat. So these recordings are from a normal behaving rat where we've implanted electrodes that allow us to drive the cholinergic inputs from the posterior hypothalamus. That in turn activates the medial septal acetylcholine neurons that then feed into neocortex, the neurons we're interested in. So in vivo, if you do this experiment, turn on the acetylcholine, you enhance any ongoing theta that was there. Then if you apply serotonin by stimulating the medial raphe nucleus at the same time to drive serotonin or 5-HT cells to cortex, you again immediately shut off the slow theta, leaving only the fast theta behind. So it's nice to have that kind of in vivo, in vitro correlation. Gives you confidence that the circuitry you're dealing with can be all in that little slice you're studying. You don't need a whole lot of ascending parts of the brain to make this kind of switch. Interestingly, when you do this in an awake animal, they immediately freeze. It's like the fear freeze signal to neocortex. It just shuts it right down with the concomitant shutdown of the slow theta. Okay, so we've got a brain slice that behaves kind of like a real animal walking around. So that's cool. We can now, get all medieval on it with microelectrodes and see if we can determine where these signals are generated and how they propagate. And we've done a lot of that work, and some of it's published. So I won't bore you with details on that. I just want you to know, for our purposes today, the theta rhythm is generated in layer 2-3 cortical neurons, and the signal then propagates up to layer 1 input layers of cortex, and then down to layers 4 and 5, which are output layers of cortex. Now, what's really interesting, at least to me, is that if you apply anesthetics to people or rats or brain slices, you produce an amazingly stereotypic response. So you have to be an EEG expert to tell the EEG effects of an anesthetic in a rat compared to a human. Almost indistinguishable. There's some subtle differences. And that's not just true for people and rats, that's true across the whole mammalian, all the different critters, cows, rats, cats, dogs, all the veterinary animals have been well studied in this regard. And you see this same stereotypic pattern of EEG effects. The most important effect for this meeting is the earliest effect of the anesthetics, which is to produce slowing of the brain rhythms. You go from the theta type rhythms and gamma and whatnot down to delta, low frequency, large amplitude rhythms. That's you'll see that in all of you. In fact, we see it in most of you every night when you go to sleep, because it's the same pattern you exhibit when you lose consciousness naturally when you sleep each night. But anesthetics are cooler, not only because you can turn them on and off whenever you want, but you can take people way deeper and do surgery on them. And so the next step down is called burst suppression patterns, and that's what you need to stop painful responses such as skin incisions during surgery. But we won't talk a whole lot about that. That's deeper than we need to be for consciousness. Suffice it to say, at loss of consciousness, you're producing a delta rhythm. I was amazed to see that same pattern occur in the brain slice. So low concentrations, you produce slowing, big amplitude delta rhythms. And then if you go up in concentration, you can produce burst suppression patterns, just like you see in an awake moving, an intact rat. So how do these rhythms slow down from theta and beta and gamma and all that down to a delta slow wave pattern? Well, it turns out that the first thing you see when you start probing the different neurons in the circuit, inhibitory neurons, pyramidal cells, input cells, output cells, they all show this same effect at the lowest concentration of anesthetics, and that's a prolongation of inhibitory postsynaptic currents. These currents come from GABA synapses, from interneurons. Inhibitory interneurons throughout the cortex are controlling the pyramidal cells. And in the presence of concentrations that produce delta slowing, you're simply prolonging the period of inhibition. You can see that at the bottom of the slide where we overlay synaptic current recordings in red with the actual rhythms, theta rhythm or delta rhythm. And you can see you just need to get to the same threshold of inhibition to account for the theta versus the delta rhythm periodicity. So that means this delta slow wave activity is intrinsic to neocortex. You don't need a thalamus or a brainstem or any other pieces of the brain because they're not connected anymore, which is a really cool finding. Now that doesn't mean that anesthetics don't affect other parts of the brain. What it's saying is for the dominant signals we record during anesthesia from the surface of the brain usually are being generated by the neocortex independent of the other effects throughout the nervous system. And there's such a great correlation between the delta rhythms and consciousness that this is a really good model for how do we go from fast type brain activity to slower rhythms that then are associated with loss of consciousness. So bottom line is you can produce these delta rhythms at loss of consciousness with a variety of different anesthetics. They're listed here. They all produce the delta rhythm at the point of loss of consciousness, whether it's in humans measured by a verbal response or in the writing reflex if you're using a rat. They're all pumping out delta frequency activity. And using the brain slice, we were able to show that this delta frequency EEG activity is produced by simply prolonging the inhibition of the inhibitory interneurons that are in turn controlling the pyramidal cells that generate the EEG that we record with surface electrodes, or in this case, in the slice as a micro EEG signal. So I'm going to end at that. There's several minutes for questions. I'd be happy to take any questions, comments, or heckles even. We're ready to fight. So I'll turn it over to you folks. Well, let's have a pop quiz first. Make sure you were paying attention. How many of you, I want you to raise your hand, how many of you are conscious? Quite a few. That's good. That's really good. How do you know you're conscious? Any ideas? Exactly. That's my answer too. I feel it. I feel I'm conscious. Now, feelings, they're not very scientific yet. Maybe someday we'll be able to measure them better, but right now they're kind of mysterious. So, any ideas how we're going to get at that problem? Can you speak up? So some form of parity between what something feels like, I guess. Oh, I'm afraid we can't hear you. Could you step to the microphone? We'll take.
So thank you very much for the nice talk. I'm Andrew Budson from Boston University and the VA in Boston. And some people have postulated that given the type of work that you've shown, that each section of cortex can produce its own EEG rhythm, that each cortical region has the ability to be independently conscious, and I just didn't know if you had any thoughts about that.
Well, certainly we've got living examples of that. So the marine mammals often sleep with one half of their brain at a time, which allows the awake half to keep them up and breathing. So there's behavioral, physiological evidence for that. Typically with these anesthetics, you produce a broad scale synchronization, and usually it's a traveling wave that progresses from frontal cortex to the posterior parts of your brain in just wave after wave of delta activity. So, but that said, there is evidence that if you have electrodes kind of scattered around you can see anomalies, regions of cortex that aren't in perfect synchrony, they may be phase shifted a little bit. So the answer is yes and no.
Thank you.
It's rare to see isolated regions doing their own thing with anesthetics. Mostly everybody's talking the same or dancing at the same beat.
Thank you.
By parity, I meant something converging that becomes more visible as you proceed. And as an example, I gave terawatt-hour production from wind turbines to seeing ice volume increase. So you have a way of measuring the parity.
Right, and certainly if that's going to be important for any of the human psychological level things we do, not just feelings, but also thought and reflection and memory and stuff like that. I agree with you that we'll have to have, we'll build correlates really between phenomena and what we think is going on in the brain.
Okay. Thank you.
Very interesting. As a novice in this field, I have a question about what you find as a consistency across, let's say, mice or even different species in terms of delta frequency or theta, especially under certain drugs. If anything has been done that way, can you actually increase the frequency? And does it increase with something like, I will take a wild guess, like dimethyltryptamine or some drugs that have opposite consequences? Is that been looked at?
Yeah, no, that's absolutely correct, Rajneesh. In fact, I showed you the one slide where if you add serotonin, you don't increase the frequency, but you block the slow frequencies and the higher frequencies persist. So you can separate these things. It turns out you can also shift them around, and behaviorally they shift around. So the theta rhythm isn't exactly 4 hertz or 6 hertz or 8 hertz. It's moving around depending on what the animal's doing or what the human's doing even. So these are not, hard and fast fixed frequencies, but they're frequency ranges, frequency bands that are commonly seen. So, and you can manipulate them in a number of ways. So for an easy way is running. If you put a rat on a running wheel, if he's walking slowly, he makes a lower frequency theta that increases along with the running speed. So that doesn't involve anything other than encouraging the rat to move faster.
Thanks.
Hi, Bruce. Appreciate your work a lot. Tam Hunt, UC Santa Barbara. And kind of a two-part question. What is the primary functional separation between slow theta and fast theta? And then second, do you see any efforts to create a concerted effort to a program to create a library of resonome frequencies with different functions across different species at this point?
Well, the first question's relatively easy to answer because it doesn't involve a lot of work. So the behavioral correlates used to be that the slow theta, the cholinergic driven one in vivo, was associated with behavior, whereas the fast theta was associated with sensory perception. But that's kind of been tossed up in the air in the last few years. It looks like there's a good degree of overlap between the so-called type 1 theta and type 2 theta. Now the type 1 theta is not cholinergically driven. We know that from in vivo studies with blockers and whatnot. The theta, the cholinergically driven theta, is associated with the motor, typically. But we now know that it's associated also with a lot of sensory integration. So it used to be pretty clear, two types of theta, different behaviors, but not so much anymore. There's crosstalk between them. Now, your second question is tough because nobody's done that. And you'd want to do it across a number of species. You'd want to do it across, to start with, a number of animals, and look for consistent patterns and peakiness. And that has not been described, but I don't think it's been well looked at either.
Yeah, agreed. And I'd love to see an effort, kind of like the Connectome project around the world, on resonant dynamics across species. Thank you.
Yeah, good idea.
Steve Dice, UCSD, retired. In sleep, delta waves are what seem to accompany glymphatic clearance. And I'm wondering if there's any clinical applications to your kind of work, whether or not you have any control over glymphatic flow and blood flow to monitor whether or not you can see amyloid products and other things being pushed out.
Well, there are commercially available EEG monitors that are used both in sleep and in anesthesia.
Mm-hmm.
The
But as far as the
difficulty is-
mechanism itself, can you see chicken and egg, whether it's the delta waves that trigger that or whether something else is triggering both?
Oh, no, clearly something else is triggering the delta waves in natural sleep. There's a number of active ascending systems that drive the cortex into the delta rhythm. Mm-hmm. And they start way down in the brainstem in various sleep nuclei. And maybe Kathleen can address that question better than I can, but because I don't know the physiology of sleep as well as I should. And it's still being untangled. It's more complicated than anesthesia because it involves several neurotransmitter systems, some turning on, some turning off. It seems to be quite a lot more complicated than just a single easy mechanism.
So you think it's neuromodulators that are triggering the interneurons to do this?
Yeah, well, that would be my guess. Yeah, I would think that's how it's working.
Thank you.
Thank you. This would have to be in human studies only, but have you experimented at all with binaural beats?
No. No, I haven't looked at that, but I think that's, there's a lot of stuff going on in the EEG that you can tease out with various analytical approaches. And I think looking for harmonics and beat interference patterns is just beginning to be looked at. As
What I'm referring to is the presentation of dichotic sounds. That is, let's say 400 hertz in one ear, 404 hertz in the other ear. So the third sound is actually produced in the brain. It mixes in the superior olivary nucleus, and a 4-beat frequency is generated, the difference between the two, and that gets sort of projected up to the cortical mantle. And there's some evidence that if you are undergoing this and you practice holding your awareness, you don't go to sleep, you don't become unconscious, but your brain does get into a 4-cycle, a 4 hertz signal, which complicates it a little bit in that,
Right.
You're still awake, but you'll fall asleep if you don't really get good at focusing while your brain is being entrained in what's called this frequency following response. But that's something you could look into. I'll talk to you later about it.
All right, thanks.
Thanks for your talk. This was kind of alluded to by the last questioner on this side, but do we have any idea, to have these these waves being produced all the time in our brain, do we have any idea what the mechanisms are that initiate and regulate and harmonize these waves, or is that still part of research?
Well, it's still a heavily investigated area of research. We know a great deal about how some parts of the brain are critical for generating some types of waves. For example, if you destroy the medial septal nucleus, you abolish theta in animals. We know that the different rhythms are generated by different mechanisms. For example, we can make gamma oscillations with selective excitatory amino acid transmitters, and theta with cholinergic. And I'm sure a lot of the rhythms will fall out being associated with different neurotransmitter systems.
But so if I could just follow up, do we have any theories about how this is coordinated and orchestrated across the whole brain? Because I'm guessing, maybe I'm wrong, that these waves are coordinated throughout the brain.
Right, and there's a couple of labs that are at the forefront of looking at that. Yorgi Busacki in New York, and I'm not sure where, I'm blanking on Brian McNaughton, and he bounces between Arizona and Alberta and somewhere else, I think Seattle. So he's got some really interesting approaches to looking at these control circuits for the different rhythms. So those two guys are where to look.
Thank you very much.
If these brain slices can undergo anesthesia, could they also be conscious? And more generally, what are your thoughts on conducting experiments on in vitro conscious brain tissue?
I'm not going to go there because I'll never get it through the animal use committee. If they think my brain slices are conscious, we won't be able to. We'll have to work on anesthetized brain slices all the time.
But isn't that a serious ethical issue, that maybe there should be a moratorium on conducting any experiments on potentially in agony brain tissue?
Well, yeah, but I'm pretty sure an isolated brain slice is not feeling anything because it's got no sensory inputs left. So now, when I stress it with neurotransmitters or electrical stimuli, then yeah, maybe that's not, it puts it in a bad mood, I don't know. But I'm not going anywhere near that because that's got philosophical as well as practical problems associated with it. As you know, brain slices are a very popular scientific tool, not just for anesthesia research, but for learning and memory and all kinds of other disease studies.
I assume it'll be a very long time before we have any means of testing whether a piece of brain tissue is in pain.
I don't know how you would do it.
No. Okay, thank you.
Okay. And this will be the last.
Yeah, so I just thank you for the wonderful talk and the research. And so I'm a quantum physicist, having actually proposed a quantum theory of the consciousness. So so in this theory, everything at the deepest level is a quantum field, and the brain and also the part of the body, physical body, can resonate with this quantum field and then receive vibrations. And this vibrational quantum field carries the information, energy, and matter. So when the body receives the field and then receive the information, energy, and matter from this quantum field, then you get this feeling, which is also connected with the consciousness. So this theory can explain why different part of the body can have, synchronicity, synchronous.
Yeah, it's like a hologram.
Yeah, like
every little piece can.
Yeah.
I'm going to save that question for Stuart when he gets here, because that's right up his alley.
Yeah, and so I'm actually interested to apply this to some of your findings. Yeah, just want to say that that's how we answer why people have the feelings, how people can have a feeling.
Oh, let's talk about that afterwards.
Yeah, and also the conscious, so that's the feelings coming from the spiritual heart with the receiver of the quantum field.
Sure.
Which is every part of the body can receive the quantum field. Yeah,
That's
so we can
possible.
talk more about that.
Okay, next we're going to have Tony HUDETZ from University of Michigan.
Check one. All right. Thank you, Bruce, and thanks the organizers for the opportunity to speak in this workshop. Thank you for this wonderful start in microscopic molecular mechanisms. I'm going to stay on the classical side of this quantum classical yin-yang. No quantum in this talk. As you can see, my title, Electrophysiological Correlates of Consciousness and Anesthesia. We think anesthetics give us a window on understanding, better understanding consciousness or its neurobiological basis. As an introduction, I'd like to point out a couple of things historically. It's a 30-year anniversary, so perhaps it's appropriate to kind of review how we got here. When I got into this, of course, the first thing that came to me was Bernard Bars's proposal. How do we study consciousness scientifically? And he said it's contrastive analysis. So we compare two different conditions or states. It's either perceived or not, or something is willed or not, or somebody is in an awake versus non-awake. But it turns out very quickly that a more nuanced distinction has to be made, because it is possible, for example, to phenomenally perceive something but not having access to that information. So there's more level, not just being aware and non-aware. And it's also possible to volitionally prepare for some action, but then reflexively execute it or even veto it. So that's another intermediate state. And then about states, you can be aware but disconnected from the environment, what we call actually covert consciousness, and so on. So it's not just simply binary comparison. This becomes actually even more clear when we look at drug-induced state changes. These are all graded. So just think of anesthesia. You can go from a completely vigilant state to drowsy to lethargic stupors, barely arousable to non-arousable and comatose, and so on. So that makes it clear that a simple binary condition, being comparing an awake conscious person to a totally unconscious person, is insufficient. If we do that, we might miss the important neural events that occur somewhere in between. So clearly we need a dose-dependent study if we want to use anesthetic to study the neural correlates of consciousness. And that's been recognized. But now, even the anesthesia monitors that are being in the operating room give you a single number, like a percentage number between 0 and 100, and that the anesthesiologist can use to get some information about the condition, the awareness condition of the subject or the patient. But as it turns out, even this is not enough, because there are further qualitative differences that have to be observed. So there could be different levels of awareness, but then there are other dimensions. And I'm just showing this figure from the Belgian group, that it is actually advisable to break up consciousness into different dimensions, where awareness is one and wakefulness is another, and behavior or connected behavior is another. There is a new article that comes out on that in JCS, should be out in the summer that I wrote. So, I like to start now with data. About 30 years ago, we talked about gamma oscillations. We're still talking about gamma oscillations. The big idea then was from Crick and Koch, suggesting that 40 hertz gamma oscillations may be the neural correlate of consciousness. This was based on other workers, of course, Lina, Singer, Talan, Baudry, Tononi, others were saying similar things. Our question was, can we apply this to an overall state such as anesthesia? And this is some early work and with Olga Imas, then postdoctoral, first grad student, later postdoctoral fellow in my lab. And we quickly showed that if we give evoke visual potential responses in a rodent by flashing LEDs in front of the eye, then we can get beautiful gamma oscillations. But as we anesthetize the rat and look at the graphs on the left, from left to right, the gaze gamma oscillations are not suppressed. If anything, these gamma oscillations get stronger. And it's important to look at whether you look at this early response of gamma within 100 milliseconds, you can do peak-to-peak analysis of the waveforms on the left, or you look at the later potentials that develop over a period of seconds. And if you look at the late potentials, it's on the right, you can see, and even here, these late so-called induced gamma activity does not diminish. In fact, it's increasing at the point where the rats become unconscious. Now, that is measured by the so-called writing reflex that Bruce was referring to. I'll get into that in a second. So even then, when we go to the transition from conscious to unconscious, these gamma oscillations are maximal. But they never really go beyond the waking baseline. Now, later on, we were able to show that this also applies to resting gamma oscillations, and at least the 40 hertz. But if you look at very high frequency gamma, which is 70, 140 hertz, then you do, in fact, see an attenuation. Then we went on and asked the next question. So instead of just looking at gamma in one position, in one place in the brain, let's look at how gamma is correlated across brain regions, one of the questions that was just asked. All these oscillations are, in fact, coordinated across the brain in particular ways. So in these subsequent experiments, we started to implant many electrodes in many brain regions, as you see on the upper left, in a rat brain, and recorded local field potentials and calculated the interaction or estimated the interaction of these field potentials over a distance using the measure called transfer entropy, which I think at the time was probably the first application of TE to electrophysiological signals, but now been used quite a lot. And so basically, in a nutshell, the result we got that yes, when the animals were anesthetized, or at least they lost their writing reflex, there was a disconnection between major frontal, parietal, and occipital regions. But there was an added feature of that, and that was that if you look at directed communication, the feedback from higher to lower brain regions were preferentially suppressed. Now, these two observations since have been reproduced in many species, in different anesthetics, and many different conditions, with some caveats. But I'd like to emphasize one thing that is not too often mentioned, that there were three critical, technically important aspects of that study. First is that we used concentric bipolar electrodes, and this is important for the electrophysiologists because that's one way to get local potentials. With EEG, you get quite a blur across the brain, but the local concentric electrode is doing something like a Laplacian transform right there without the calculation. So you get highly local, And so you can calculate connectivity between these. The other thing is the use of a sensory stimulus, not just resting activity. The sensory stimulus resets the oscillations in the brain. So you get a time reference to which you can calculate and average, if you wish, the evoked potentials. And the final important thing was to use single trial analysis. And that is because as you get further away in time from the original stimulus, the waves begin to disperse. And so if I simply average the waveforms, I lose the signal. So single trial analysis got us over that. Okay, enough about that. I want to mention actually the work of my good colleague Dinesh Pal. We jointly published this paper with him that it is possible, though, that sometimes these gamma oscillations do not strictly follow what we expect. So here, Dinesh used a pharmacological effect infusing carbachol into prefrontal cortex or parietal cortex, trying to awaken the rats from anesthesia. And this was successful in many rats. And it was also shown that this kind of the transfer entropy in the gamma range that I just demonstrated, which we found earlier, was indeed reduced under anesthesia. In this case was sevoflurane. However, when the rats were woken up behaviorally, the gamma did not come back. And this was true whether the rats woke up or not woke up. So it is important that as we probe the brain more and more in more detail, we want to decipher which pathways are responsible for going under or recovering for anesthesia. We are modulating specific pathways. And some of the pathways we modulate by locally administered drug do not affect the globality, the global workings of the brain. Some phenomena are selectively can be altered while others are not. I have a few more general comments before continuing with the data. And this is something that always I think about with these kind of experiments. The two notable complications. One is that we are not quite sure what kind of relationship there should be between the level of consciousness and some kind of neural activity in the brain. So it could be any measure of consciousness level or any measure of neural activity. The relationship could be very dramatic or abrupt, that's the red line on the left, or it could be gradual. Why is this important? Well, if there is a definite neural activity that correlates with loss and return of consciousness, well, let's call it the NCC for unconsciousness, it's a very definite and abrupt transition, we can find that. But if the relationship is very gradual, then we won't have an extra information to know where on that gradual transition, what are we looking for, where the critical neural events occurred. So this is one thing that is still an outstanding question. The other one is the history. This is well known in electrophysiology circles who study these phenomena. And that simply means that if I induce anesthesia, I see neural activity change, and I see animals or humans lose consciousness at a certain level of intervention, perhaps at some level of anesthetic dose. But if I remove the anesthetic and I allow neural activity to recover, it may be a different level at which consciousness recovers. So what that means is there may not be a unitary NCC for loss and return of consciousness. It will depend on the route or the pathway you get to that, whether increasing or decreasing. So let me skip the next one and continue. Yeah, yellow light. Okay, one more conceptual slide. So where should we look for the NCC? I mean, what organizational level? So obviously, you can think of consciousness as a holistic thing. And it's actually appealing to say, well, consciousness is produced by the whole brain. It's a global, holistic phenomenon. But you can also have the local views, where you depend on you're really addressing detailed synaptic and molecular effects. I'd say that it depends on your methodology. If you're a brain imager, you probably will be looking for the NCC and regional and global level. If you're a chemist, you probably will look for the answers in molecular mechanism, and so on. In any way, we need all of these. So let me go on. So we continued our work here about studying, going down from local field potentials to spikes. So why spikes? Spikes carry the information in the brain. Local field potentials give you very useful information about population activity, and it gives you some window on dynamics, which is all very important and useful. But if you want to understand how individual neurons communicate, you actually need to look at their spiking activity. And not only what their average spike rate is, which you see, by the way, two examples in the middle. The top shows 4 neurons spiking along happily when a rat is awake, and below that is when it's anesthetized. And you can see the pattern of changes. So that is very obvious. And we know, by the way, when the rat is anesthetized, as Stuart said this morning, the brain is still working, but it's only working at half the speed. All the neurons slow down. So it's not quite that everything is fine and dandy. There is definitely a slowing and alteration in neural activity pattern throughout the brain. This is from visual cortex as an example. But rather, the question is, how do neurons communicate? How do they send spikes to each other? Or how complex their interaction is? This can be measured by some suitable quantities. I use in this case one that was introduced by Tononis, the CX on the lower right, this formula based on the entropy of the spike patterns, and it's called neural interaction complexity. And in the middle is the important result, which I'd like to explain, and that is how this complexity measure of neurospikes changes with the anesthetic dose. And here we use desflurane, a gas anesthetic similar to isoflurane that needs higher doses to work. Again, we use the light LEDs to elicit visual evoked potentials, and we evoke the complexity before the LED light and right after the LED light. So right after is a visual evoked complexity measure. So that's the red, the post, the red line, and below is the blue line. And it's very clear that as sensory stimulation increases the complexity of neural complexity, to be short, at every anesthetic level, including wakefulness, which is 0%. However, there is an important difference in the characteristic of the way these curves change. And if you notice, the gray zone is where the rats would lose their writing reflex, somewhere between 4 and 6%. They fall off their feet. They don't want to move around. They don't want to regain their posture. And so the first 3 points are fairly steady. All these states are conscious, according to our presumption, if the writing reflex measure is correct. Conscious, but we start to see a drop in complexity just when the writing reflex goes away. So this is important because it gives us a threshold phenomenon. If you think back what I wanted to explain here about the abruptness, at least this gives us some measure that indeed gives us a threshold of transition where we can say, yes, this is perhaps an NCC exactly for when consciousness is lost. All right, I'm still in time. All right, another important thing is to understand how brain arousal affects these processes. So that, in a nutshell, in this experiment, we continue to look at complexity, and now we add a stimulus, a brainstem stimulant, electrically stimulating brainstem in the nucleus pontis oralis in the upper left, you can see that. And we still have the visual evoked potential, but now we're recording in higher cortex. So far, everything I've said was in primary visual cortex. But from point of view of consciousness, we think the higher order integration is going to be important. And what happens in higher cortex, association cortex, both in parietal, frontal, and other regions. So here we went one step further. So we're now looking at secondary visual cortex, V2, as well as parietal association cortex. And what we're able to show that once you stimulate electrically the ascending arousal system, you get immediately a very big amplification of sensory signal processing. And that is, it's a complex figure, I understand. That is visible in the green bars and dark blue bars in the very center on the left and the right. And this is done at 3 different levels of anesthesia. The 3.5% on top is very light. So these rats at 3.5% desphorine are still conscious. They're very close to losing consciousness. But then as you go down to 4.5, in particular 6%, they definitely out. So you can see that even in those conditions, the arousal system can restore or largely augment the sensory processing, which makes actually the conclusion somewhat interesting, because you ask yourself if there is sensory processing in higher cortex, is this animal conscious of it or aware of it? Now, we know the writing reflex is gone, at least at 4.5%, so they behaviorally don't show us any sign of being ready to jump up and run away, but their sensory systems are working actually supranormally. So I don't have a final conclusion of that in terms of explaining it, but I want to bring it up that it's a difficulty that we need to deal with, and philosophically, I guess, understand where the critical neural phenomena are that we can relate to the conscious state. Okay, so one more interesting question that there are, of course, these theories out there. What kind of large-scale communication in the brain is important? For example, we showed that anesthetics suppress the feedback process from higher order cortex down to lower cortex, all the way from the front to the parietal to the occipital. But does this happen also locally? So we investigated that recently. And instead of local fit potentials, we actually use spike transfer. Now, the spikes, as I said, are very informative, but it's very difficult to get very long-range connections for spikes because it's so difficult to find the corresponding neurons that talk to each other. But for a shorter range, like visual 1 and visual 2, primary visual and secondary visual, you see that in the center, you can do that. So again, use the transfer entropy measure, and we use an electrode that ends up in different depths, so supragranular and infragranular. and they're alternating. You can see the arrangement on the right. So we measure from supra and infra from many, many regions simultaneously, and take advantage of the fact that the feed-forward and the feedback signaling has very specific anatomical arrangement. So the feedback goes from more supragranular regions to, and mostly infragranular regions. I'm sorry, I started with the feedback. Infragranular regions to both supra and infragranular, and the feed-forward goes mainly from supragranular to the middle layers, the input layer. So with this recording, you can map these connections, and what you find is that there is indeed a fairly large reduction in communication between primary visual and secondary visual cortex. This is a dose-dependent effect you can see in the middle with the desflurane concentrations. 8% is actually a very deep anesthesia. It's already burst suppression Bruce was talking about. And then when you reduce the anesthetic, so we see the reversal side, we see that everything comes back. Interestingly, much higher actually than upon awakening, than before induction of anesthesia. And we also see that the feedback is more reduced than the feed forward as a right panel of the thing. So this actually was predicted by Victor Lam's group, that local feedback is part of a hierarchical, or local feed forward and feedback, so recurrent processing. Local recurrent processing is part of a hierarchical system that not only long-range interaction occurs, but regions to regions. And all this is necessary for conscious sensory processing. Right. Okay, so the last three works I'd like to talk about, this is very recent. So my former colleague, Dr. Han-Soo Lee, postdoctoral colleague, discovered this phenomenon called the paradoxical state, or paradoxical activation in anesthesia. So where this comes from is, I always thought, and probably most people think, that if I give a gas anesthetic at a fixed level, that will, of course, result in fixed concentration in the lungs and in the tissues, including the brain, that the anesthesia would be stable, a steady state. And I can dial in very easily different anesthetic concentrations, and that would hold. But as it turns out, there were a couple of observations in humans, also from our group, that despite of a constant alveolar concentration of anesthetic, the states are fluctuating. Of course, we know during natural sleep, you have different sleep phases, and they spontaneously change. But in anesthesia, it's supposed to be constant, and it's not. So we wanted to investigate this with spikes. What happens at the local level? Because one thing, you saw some signs of this in EEG, but what do the neurons do? So it turns out that there is something dramatic that happens in deep anesthesia. And if you look at the color figure, or the top figure, we divided brain states into 5. We've actually found 5 different brain states. And the anesthetic concentrations shown on the left with the, I don't know if you can see my, yeah, you can see my pointer. Yes, this red line shows we starting at higher anesthetic concentration. We step down and step down and step down. And the spectrogram is a local field potential spectrogram. And up here we have spike resters and also local field potential. So what I want to call your attention to is this change here. So this is the deepest level of anesthesia. This is 6%, basically, all along. But there are 2 very different states. This one is normal deep sleep, slow waves, as Bruce explained this morning, slow wave state. But this is a totally activated period that almost looks like an awake. So how does that happen that during deep anesthesia, all of a sudden you get an EEG, local field potential, and also spike activity that looks very nearly same as awake. In this sequence, this is actually state S3, arbitrary. It's the third one. This is deep anesthesia, burst suppression. This is a transitional period. This is a lighter state. You see slow wave, and this is awake. And this S3, this paradoxical state, is actually much lighter than what you expect from the given anesthetic concentration. And if you look at the pie charts, it shows you that the teal color, which is this paradoxical state, occurs mostly in the deepest anesthetic state. There's something that happens in the brain, the deeply anesthetized brain, that throws it back spontaneously to an activated level. And this reminded me years ago, French neuroscientist Alain Dexax had an article in which the title said, "Are corticothalamic upstates fragments of wakefulness?" So these are here on the left, downstates and upstates. So how do you know that? Maybe you can wake up during these anesthesia for seconds or maybe even minutes. So my colleague, Dr. Duan Li, research scientist in our department, has now looked at this more carefully. And we asked the question, we see there's some activation in neural spike rates. What about the complexity or the recently introduced the PCI index, which is perturbational complexity index, introduced by Tononi and others? How do those change? They may be more sensitive indices. So without going into too much technical detail, I can tell you the answer. The answer is that the paradoxical state is still an unconscious state. If you look at perhaps the panel I'm pointing here with my cursor, that shows that as anesthesia goes on, the complexity of the paradoxical and the PCI of the paradoxical state is indeed reduced. So I have 6 minutes left. I'd like to complete with a couple of closing thoughts. So we see that depending on whether we measure global signals or population signals or spikes, unconscious has many signatures in spectral changes, connectivity, and complexity. What we are not yet sure is any of these are causally related to loss and return of consciousness. Some of them could be prerequisites, other could be consequences. It's an unsolved question. We definitely continue interested in neural events that strictly occur at loss of responsiveness and return of responsiveness. And we have found one of these is the complexity, in neural interaction complexity, as I showed earlier. Have to admit the caveat that behavioral assessment is still a limiting factor in knowing when animals or even humans lose consciousness or recover consciousness. Because covert consciousness in loss of responsiveness cannot be excluded. So another question, a final question, is everything I've shown was in cortex. But is it only cortex that matters? And most of the NCC studies, of course, have been on cortex and thalamocortical systems. But now, subcortical systems are also quite important. They intertwine through the arousal system as well as other ways. And just an interesting thing, Jeff Hawkins, who's author of 2 very, very influential books on consciousness, he said, he was asked, what is consciousness? And he said, well, consciousness is like having a cortex. This is kind of interesting, but I think it's more than cortex. It's a lot of things in the brain that have to work together, and more to come. So I want to thank 2 more people who in the lab who I didn't mention along the thing. Dr. Wang, who is an expert neurosurgeon, and Kathy Zelanak, who keeps our operations going. Thank you. See, I have 4 minutes left.
Thank you. Can I ask, do you know what happens in sleep deprivation to the EEG, to the recordings?
I have not recorded anything in sleep. I'm sorry. I have colleagues who do a lot of their living for sleep, but I don't think they calculate complexity or this kind of stuff. So that's to be done.
May I ask, sorry, this side? Hello? Sorry, this. You want to go first?
Okay.
Oh, okay. It's very interesting. It's something that I've been pondering myself. I think when we think about consciousness, we have certain ideas that, like behavior and these things, have to be present. What if function, even if you go to slices, perhaps even a slice or different organs have functions, and by loss of consciousness, what we mean is loss of those functions. When they all are integrated into the cortex or in the brain has that function. So when we are studying consciousness, perhaps we are focusing on that integration process, and we are missing the individual functions that different organs and different parts may have. That also gets around the aspect of pain and many other things that slices or may not. And I think your study shows, in a way, that there are like layers and layers of functionality, perhaps, that are being suppressed, and then integration is a separate issue.
Are you thinking of behavioral inhibition or neuronal?
Neuronal.
Inhibitor.
Inhibitor.
Yes. So I did not have time to talk about that, but anesthetics have, of course, very selective effects on inhibitory neurons and excitatory neurons. And the interesting thing about that is that when you study that in vitro, they're very specific. They augment inhibition in particular and suppress excitation. But in vivo, in the intact system, this actually doesn't play out that way. In fact, the excitatory drive is suppressed more, and the inhibitory drive stays constant. It's hard to predict from in vitro. That's not exactly the answer to your question. But yes, we are looking at separately also the complexity of the inhibitory network and the excitatory network. Can separate those. And anyway,
but
the more answers will come.
So that might suggest that the inhibitory and excitatory networks, they're responding to some stimulus, and the anesthetics are suppressing the ability to respond, which means that at a higher level, it's the integration of all this information. Whether that plays out in vivo, that's a good question.
Yeah. Yes, thank you. Thank you. Good comment. Thank you.
Thank you for your talk. If I understood you correctly, I thought you were indicating that under anesthesia and when the subject is unconscious, there's still responsiveness to visual stimulation. Does that possibly mean, like, have there been any studies to see if a human who's under anesthesia and unconscious could learn or be told or taught things that would then be recoverable post anesthesia?
Yes, what you said is correct. From the spike analysis, we see an enhanced visual response in V2 and parietal. But as far as further processing, our group is also conducting fMRI studies in humans, and Dr. Huang will present Saturday his work on that, and that might include some of those that we have tested humans in under anesthesia, asking them to do mental imagery and that sort of thing. So it's not strictly visual, but rather, and that is ongoing work. And we had some very small success in showing 1.5 patient, 1.5 subject was able to show, but very low rate, but that's ongoing.
Thank you.
I think
Oh, sorry.
it was a fabulous talk, Dr. HUDETZ. Thank you. I was really interested in the perturbations of complexity. That seems to me an amazing metric that you're pointing to as a kind of a threshold for consciousness. And I wonder if you could talk more about that. Is that an indication, for example, that self-organizing mechanisms are suppressed? How does this impact, transient dynamics as well as stable heteroclinic chains, fractal noise?
Yes, so complexity, of course, has many metrics, and there are many alternatives, and we do not know what's the best metric. The best metric, well, some metric comes out of information integration theory, of course, that some is not computable, but there are other things. The one we use is a simplified computable metric. But I agree with you, the critical question will be to better understand what kind of complexity it measures, what does it mean functionally. And I have the feeling that some AI work may be actually helpful here in artificial neural networks, that we're actually looking into that, to create functional states in SNN, spiking neural networks, and then ask the question what kind of complexity they acquire if we precisely define their functional state in terms of information processing, ongoing work. So I think answers will come from something like that, modeling type work. But we're still working, and our colleague, HYUNWOO Yang, will actually talk about that in the next talk, or the talk after, some more in human context. I think I'm out of time. I don't know, Bruce.
One last question. Thanks for the talk. I was just thinking about dissociative anesthesia with ketamine and the EEG data with that, if there is any, and also the impact of post-ECT altered conscious states for people having psychiatric illness post-treatment. During ECT administration, there will be an EEG collection as well, usually. So have you got any data or insights about that?
Yes, so there are data with ketamine. I did not personally, but our neighbor lab, Dr. Dina Spal's lab, has done comparative studies, and other colleagues. Ketamine definitely produces different state. It also increases complexity, but it is, and it generates, and it also reduces feedback signaling, similarly to other anesthetics. But there are still some additional qualitative differences in its action. And I think that, as much I know, this needs to be further mapped out to what brain regions. We've been very focused on visual and nearby regions and now prefrontal, but I predict there'll be substantial differences between psychoactive drugs and ketamine, nitrous, as well as more traditional GABAergic agents.
And with the electricity impact, post-ECT because it's a lot of changes in physical systems. Post-electroconvulsive therapy, ECT.
Yeah, that I cannot comment on. I don't have any experience with that.
Do you have time for one more? One more? Yeah?
Yes.
So I'm intrigued by the work you talked about in the beginning, looking at gamma band impacts from anesthesia. Have you done similar work looking at theta? Because theta, of course, is one of its roles is to connect distant brain parts. So in the thesis that complexity and connectivity is perhaps the key process NCC for mammalian consciousness, has anyone looked at theta as the key process?
Well, Dr. MACIVER did a lot of work on theta, so we didn't want to take his fame here. But actually we did some studies with cholinergic activation, for example, physostigmine administration, neostigmine, and other means to reverse anesthetic state. And that's going to be subject of a next talk, I guess, in part. And what we saw is that in vivo, with cholinergic activation, you get a very defined, very strong theta in prefrontal cortex as well as in a hippocampal cortex above the hippocampal region. We have not looked at complexity measures of that. One could look at that. That could be interesting.
Thank you.
So our next speaker is going to talk about how to recover from anesthesia and how to promote recovery in anesthesia and what are the brain mechanisms involved in that. So go ahead, Kathleen, whenever you're ready.
Thanks so much. I'm Kathleen. I'm an instructor in the Department of Anesthesia, Critical Care, and Pain Medicine at Mass General and Harvard Medical School. And instead of talking about anesthetic-induced unconsciousness, I'm going to be speaking more about the process by which the brain restores consciousness following general anesthesia. We're really talking about it from the perspective of preclinical animal models and looking at brain regions and some circuits that might be involved. So I think by now we know what general anesthesia is, but some important features are it's drug-induced, it's reversible, and it consists of unconsciousness, amnesia, analgesia, and akinesia, which is lack of movement. So two important characteristics, I think, is one, it's got to be reversible, and surprisingly, we don't really have any approved drugs to actively reverse the process of anesthesia. You just have to wait for the drug to be metabolized. And the other one is unconsciousness, which is presumed because people don't have a memory of awareness during general anesthesia. And since this is a consciousness meeting and my first time here, I want to make sure that I'm a little bit clear on what I'm referring to with these terms of consciousness and unconsciousness. And it's kind of been presented in the talk before me, but I only have two dimensions in my graph. But one dimension is sort of this level of consciousness, that arousal, and the other being concept of the content of that consciousness, with conscious wakefulness being pretty high and going decreasing along both those axes as we get deeper into deeper sleep. You have characteristics where there may be more content in consciousness without as much level, or states like minimally conscious states where there may be some responsiveness to the environment, but we don't believe there's much going on. And a goal of anesthesia or general anesthesia is to really minimize both those dimensions of consciousness, which I think makes it a really great tool to understand how the brain gets from this really low level of consciousness back to a normal waking state. And it's not only like interesting from a neuroscience perspective, which is kind of the place where I come at it from. It would be useful because if we understand how the brain restores consciousness, we might be able to develop tools or drugs in order to facilitate that process when people don't recover consciousness as well. But that's a complicated process, in part because general anesthetics are pretty dirty drugs. I took this table and adapted it from the 2008 Science article. And it just sort of illustrates the diversity of receptors and ion channels that different anesthetics act on. Not only are there a lot of receptors and ion channels, they're not necessarily all being acted on in the same direction with the same intensity. And so normally you would probably try to reverse a drug by reversing all of its effects on its receptors. And with general anesthetics, this is a bit more challenging. So instead, I come at it from the perspective of let's try to activate arousal circuits, of which there's many, and there's several preclinical and clinical trials looking at these different pathways in order to rescue consciousness following general anesthesia. One is adenosine, right? You can inhibit adenosine receptors with caffeine, and there's a lot of preclinical studies on rodents and into clinical trials using caffeine to facilitate conscious recovery. There's acetylcholine, a lot of great work, which some of it was done by Dinesh Pal, showing that cholinergic activation from the basal forebrain into the prefrontal cortex can be used to restore consciousness in rodents. There's the orexinergic system, also known as hypocretin, which when it's dysregulated, you get narcolepsy. There's actually some drugs that are targeted for people with narcolepsy being used to reverse general anesthesia. And two that I'll be talking more about today, because this is really the focus of my research, is the norepinephrine system, which largely produced by the locus coeruleus, this region in the brainstem, and dopaminergic system, which is probably more well known for its role in reward and arousal. Now we have two great pharmacological tools that we can use to modulate dopamine and norepinephrine that's already approved clinically. We can do this by modulating the reuptake transporters for dopamine and norepinephrine. We have methylphenidate, which is also known as Ritalin, used for treating of ADHD, and that blocks both of those transporters, so you get this increased presence of dopamine and norepinephrine in the synapse, so you can have it consistently activating on postsynaptic receptors. And a related drug, D-amphetamine, also an ADHD medication, which not only prevents these dopamine and norepinephrine from being taken up into the cell, but also pumps them back out, so it's a pretty profound effect. It was about a decade ago where my mentor, the lab who I work in, Dr. Ken Solt, used Ritalin and amphetamine intravenously to show that you can deliver this in animals under continuous general anesthesia to actively reverse it. And here's what it basically looks like. These are two rats. They're inhaling sevoflurane, and the rat closest to us just received an IV of D-amphetamine, and they're both getting a saline flush afterwards. You can see they're on their back. That's that loss of the righting reflex. It's what we often use as our general proxy for loss of consciousness. And even though this animal that's closest to us is still inhaling the same concentration of sevoflurane, has now been able to roll over and start making movements. And that goes along with some EEG features of wakefulness, with D-amphetamine producing this awake-like spectrograph changes. And even though amphetamine causes an increase in both dopamine and norepinephrine, you can entirely block this effect, both the EEG signatures and that behavioral sign of arousal, by just blocking dopamine receptors. So we believe that this effect is at least driven mostly by dopamine. But there is more evidence suggesting that it is a dopamine effect. There is another drug, atomoxetine, and you can use this and it only blocks the reuptake of norepinephrine, so dopamine concentration shouldn't be directly implicated in the application of this drug. And you can see from the EEG, you still get this rescue of awake-like EEG signatures with atomoxetine, but none of the animals actually woke up. So it's very similar to other work by Dinesh Pal showing there's a dissociation between some EEG features of wakefulness and behavioral signs of arousal in the rat. Now, since those initial studies, methylphenidate and D-amphetamine have been tested under different kinds of anesthetics. So it's been shown to reverse isoflurane, sevoflurane, propofol anesthesia in rodent models. Those, although they target multiple receptors, as I said, the main one that is thought to contribute to the loss of consciousness is the GABA-A receptor. We can also use it to reverse unconsciousness induced by fentanyl, right, which is a mu opioid receptor antagonist. And also, most recently, it can be used to reverse a drug called dexmedetomidine. Dexmedetomidine is more of a sedative. It's usually used in conjunction with general anesthesia, but you can give it in sufficiently high doses that animals will have a very prolonged loss of writing. And it works by activating the alpha-2 adrenergic receptors, right? So it prevents norepinephrine from being released from the locus coeruleus. And just like the rest of them, you can block that effect entirely with dopamine receptor antagonists. The one exception to this rule is ketamine. Ketamine, where at any dose that we've tested these stimulants, you can't change the EEG signatures and you don't produce any behavioral signs of arousal. So a bit more on this later. But so dopamine acts on or has many different pathways by which it exerts its effects. Some quick review of the dopamine pathway. There's a mesolimbic pathway. It starts in the ventral tegmental area. It's the midbrain region and projects the nucleus accumbens. It's really thought to be involved in reward. The mesocortical also starts in the VTA, but projects the prefrontal cortex. Nigrostriatal is more the substantia nigra to the dorsal striatum. That's the one that when you have a loss of dopamine, you get the Parkinsonian-like symptoms. And the last one's the tuberofundibular pathway, median eminence to the pituitary gland. So it's more endocrine function. So to try to find out whereabouts in these regions we're seeing an effect of dopamine being able to overcome general anesthesia, we implanted these deep brain stimulating electrodes into the ventral tegmental area. So it has many dopamine neurons. It's not exclusively dopamine. But with that, we can do some studies that you couldn't normally do on people and stimulate animals under continuous high doses of general anesthesia. And so here's what the experimental setup looks like. We have these animals, they're unconscious, they're being delivered different kinds of anesthetics through intravenous or inhalational. We're continuously recording the EEG, and then we can turn on the stimulus after we expose them to the anesthetic. And this is an animal under dexmedetomidine. The stimulation has been turned on. She immediately rolls over and starts walking. And we turn the stimulation off right after. And within a few seconds, we can actually flip that animal back on her back, and she will stay unrighted for an hour or two. This is how high the doses we're using. And so really we have this very quick switch from on to off of consciousness by stimulating this VTA region. This also works very well with propofol and sevoflurane that's been previously published. It also produces this transient change in the EEG signature, so you can see with DEX, high power in the low frequency bands, and stimulation is a very transient perturbation of that EEG signature that rapidly returns as soon as we turn the stimulation off. What was actually surprising for us that we wanted to be able to have a method of reversing quite a diverse range of anesthetics, and fentanyl not used as a general anesthetic for obvious reasons. We deliver it in high enough doses to rats where they survive it, but they go completely rigid, and you can flip them on their back, and they'll stay like that for 20 to 30 minutes in this hypoxic state, and showing EEG signatures of unconsciousness. And so you can see I'm turning the animal on its back, and it's pretty much frozen as like a rat popsicle. And at 25 minutes, you'll see what happens when we turn that electric stimulation on. The animal returns to her feet, starts moving around again, and as soon as we turn it off, the movement stops. But more than that, you actually see the rigidity fully returning. So this suggests that the stimulation that we're doing is occurring with that drug on board. It's still acting on the brain, it's still exerting its effect, but you can overcome it with sufficient stimulation of this VTA area. So we've tested this with a wide range of drugs, like I said, sevoflurane, dexmedetomidine, fentanyl. And the one standout, once again, is ketamine. Even with this very potent stimulation, these animals largely do not recover. One animal righted with the highest current, but this was an anomaly, not the rule. And just to prove that this is not just an effect of stimulating anywhere in the deep in the brain, a lot of these experiments happened when I was first learning how to do stereotaxic experiments, and not all of my electrodes wound up in the VTA. And so several of them end up migrating to other areas, and when we put in an electrode, we cover it in dye so we can see where it ends up. And any time an electrode was well outside the VTA, none of these animals woke up with any amount of stimulation. So it did require that it was at least in our area of interest. Now, like I said, the ventral tegmental area has a lot of dopamine neurons, but it also has glutamate neurons and GABA neurons. We can't really distinguish cell type with this method. But we have a great tool in order to investigate these circuits in animals. So it's called optogenetics. In it, I use a targeted virus. So we use a tyrosine hydroxylase promoter. It's important, or it's required for the synthesis of dopamine. And that promoter drives the expression of this ion channel that's sensitive to light called channel rhodopsin. And that ion channel is coupled to a fluorescent tag so we can see where that virus ends up. We package all that into a virus, inject it into the VTA, and that way only our dopamine neurons are now expressing this unique little ion channel. And we can place 2 optical fibers on both sides of the ventral tegmental area right above where our ion channels are expressed. And normally, the channel rhodopsin channel that I'm expressing in the VTA, it's closed, nothing happens. But in response to blue light, it opens and you can dictate the firing pattern of these neurons. And so these experiments are currently underway with testing a lot of our standard different kinds of anesthetics to see how they can be reversed with dopamine selective activation. And here's a brief video of a rat who has 2 fibers in the VTA, and he's currently under a continuous dose of sevoflurane. We wait until the brain becomes very saturated with that level, so we're constantly measuring the amount of sevoflurane in the chamber. So you can see the blue light is turning on. It's activating only those dopamine neurons, and even though he's still breathing in sevoflurane, he starts to try to ambulate, try to flip himself over. He's a little bit big for the box, but once he does, he's very committed to attempting to escape shortly thereafter. And along with this photostimulation, where we see that behavioral activation, we also see a change in the EEG signatures. So under sevoflurane, it's that slow waves, and with the blue light on, you get a return of a more awake-like baseline EEG signatures. And so that's really identifying the dopamine neurons in the ventral tegmental area as a potential way to kickstart the brain back into a more awake-like state, even under continuous anesthesia. So one of this great applications of optogenetics is you don't have to just shine the light where the cells are. You can move the light over to downstream pathways because these ion channels are expressed all along the axons, and you can assess what the contribution of selective projections are. And this has been really a very quickly emerging body of literature in the anesthesia field, where it's been mapping out the different subcortical, cortical circuits that can be targeted, activated in order to facilitate recovery in animal models. And this is just a sort of a brief snapshot of what the research looked like when I was originally putting together this graph probably about 4 months ago, so it's already out of date. But the optogenetic studies in rats and mice have really identified that not only are there a lot of different circuits and subcortical structures that can be targeted to facilitate that recovery, but they can involve glutamate, dopamine, orexin, acetylcholine, norepinephrine, serotonin. So we're really getting these detailed maps that we can use to induce emergence following general anesthesia. It should be noted that most of these studies are done on isoflurane or sevoflurane, sometimes propofol. Those tend to be more the GABAergic anesthetics, so few of them look at ketamine. All right, so far, I've been sort of speaking about this one dimension of dopamine maybe being able to promote an increased level of arousal by measuring either this writing reflex, which you may not believe is really conscious, or some very simple EEGs. We don't do too complicated EEG structures here. But To get to the content of consciousness with animals, it's a bit challenging, and you may wonder with all these videos that you're seeing these animals writing and making kind of random movements are they actually there? Are they cognitively aware? And I can't tell you what their awareness is, so instead, to understand that second dimension, I'm really going to be talking about behavioral correlates of cognitive function, which is a bit of a cop-out. These are not identical endpoints, but I think that these tools that we can look at cognitive function can at least tell us a little bit about what that recovery process looks like. And the way that I look at it is through operant conditioning paradigms. So I'm really interested in sustained attention. In it, I study it by training animals to play this kind of a boring video game. It's called the 5-choice serial reaction time task. So we have these chambers. On one side, we have a food tray that dispenses pellets as little rewards, and the other side is the tablet. And the tablet has 5 windows, and on those windows, one of the squares will light up in a pseudo-random fashion after the animal initiates a trial. If the animal gets it right, they get a little reward as a food pellet, and they learn to play this game. This is kind of what it looks like from above. This is an animal. She just initiated a trial by poking into the food tray. You can't see the stimulus from this angle, but she did a nose poke into the correct hole. She got it correct. She goes back to get her food reward. If she initiates a trial and is distracted, not paying attention, she only has 5 seconds in order to make a selection, in which case, if she doesn't make a selection, it's considered an omission. She gets a timeout for 5 seconds, the house lights turn on, and she has to wait for the next trial to start before she can get a reward. If instead she's making a random selection to the touch screen, it's an incorrect response. Once again, the house lights turn on, and she has a 5-second timeout. And so with this, we can train animals to become very good at this task. Rats are very intelligent animals, and they're very happy to work for treats if you food deprive them. And so with this paradigm, I was able to train animals and get a sense of what their baseline attention level is day to day that they're very willing to do and show me. And once they're fully trained, we can use this paradigm to ask questions about what their cognitive function looks like as they're coming out of general anesthesia. And this came on a few years ago now, but we tested different general anesthetics. They were exposed for a period of time enough to saturate the brain for the volatile anesthetics, or enough to produce a prolonged loss of consciousness or loss of writing for the intravenous anesthetics. And we placed them unconscious in the chamber. And the first thing we do is we wait to see how long before they can write, which is our proxy for return of consciousness. And the second one is how long before they can start interacting with this game in a way that is similar to their baseline. At what point is their mind cognitively at their pre-anesthetic level? So on the left is a video of a rat following isoflurane and recovering from 1 hour of inhaling isoflurane. And after she writes up, she tries to initiate a trial, but initially there's some motor problems that are impairing her ability to play the game all that well. But even after her motor performance recovers, there's some challenges in making all the trials either correctly or avoiding omissions. But by 18 minutes, this animal was back to her baseline performance. She was getting them right, and she was missing relatively few trials. So at this point, we can kind of say, okay, this animal is, cognitively back to their state where she was before she received an anesthetic. In contrast, some anesthetics that I tested showed a really poor recovery rate, even several hours after they recovered writing. And one case of this was dexmedetomidine, our sedative agent. Even an hour after the animal writes, they do try to do the task. They initiate trials, but they will have this fluctuating attention where they can't make selections in time. Or when they do make selections, they're at random. And we only test them for 3 hours. Any more than that, they would get dehydrated. But important to say that all the animals are absolutely back to baseline by 1 day after any of these anesthetic conditions. And what these results showed us is one thing was that you can measure cognitive recovery in this sort of a continuous way following general anesthesia, and that some of these anesthetics that we tested, like isoflurane, sevoflurane, and propofol, they have a relatively quick recovery rate. So to read these graphs, the x-axis is how long have they been awake for, as measured by the return of the writing reflex, and then it's just how long before they hit that criteria that I set for can they perform this task again. And other anesthetics, ketamine and dexmedetomidine, were very challenging for these animals to recover. They're in a state where they can't play this game very well. But another finding that was surprising was that how quickly animals recover writing had absolutely no bearing on how quickly they could recover performance on this task, which was to me a bit surprising. I assumed that these would be along the same continuum of recovery, maybe related to the elimination of the anesthetic. It also had no bearing on how good these animals were at the task before getting the anesthetic. So we're really measuring two different things now. We can measure some amount of level of consciousness with our writing, and the cognitive recovery is possibly driven by something different. Now that really leads to the questions, well, the question that I'm interested in, can we actually improve that cognitive recovery, particularly with the tools that we're already using to try to facilitate conscious recovery? And to do that, we're using a similar technique as the optogenetics. Instead, it's chemogenetics. So optogenetics is great for very transient activation at a specific frequency. Chemogenetics, it allows you to have prolonged activation for several hours, which is kind of ideal for assessing cognitive function. So how it works is the chemogenetic receptor is called a designer receptor, exclusively activated by a designer drug, or a DREAD. It's also packaged into a virus, injected into the VTA, so we can express this unique receptor in our dopamine neurons in the VTA. That receptor is a G protein coupled receptor. It has no endogenous activity, so it just sits there silently until we give a designer drug. It's called clozapine N-oxide. And once you deliver that drug systemically, it will go and target these G protein coupled receptors, causing this prolonged activation. So you can see what it looks like to boost the activity of dopamine neurons in animals following general anesthesia. Now, I've only tested this in 2 different anesthetics that showed a poor recovery profile with it. The fast recovering anesthetics, the animals are already too good at the task in order to really see improvement. So this is what it looks like to test the effect of dopamine activation in the VTA following dexmedetomidine. So on the left, this is the same animal. She's tested about 1 week apart. And on the left, she's given saline, and on the right, clozapine N-oxide. So that's the one where we're activating dopamine neurons. And they write around the same time, and they're both pretty terrible right after they've writed. But by 30 minutes later, the one that has activated this reward pathway, this dopamine pathway in the VTA, she's already back to her baseline behavior. She's able to do this task very well. She's getting them right. She's missing very few trials. Whereas a saline-treated animal, she looks like a pretty standard dexmedetomidine-treated animal, having a very slow recovery and poor cognitive outcomes. So what does this look like overall with those studies? Well, clozapine N-oxide, activating the dopamine neurons, they were more likely to recover following dexmedetomidine, and when they do recover, they do it more quickly. Now, the obvious question is this, I mentioned a few times, dopamine in the VTA, it's very known for being a reward pathway. Am I just increasing the saliency of that reward, or is this an attentional effect, or is this some other byproduct? And we can't test that exclusively. It's hard to disentangle these components because it's likely a combination of all of them. But we do have some metrics that this task provides us that can give us an indication of things like motivation, motor control, and mobility to see if these are independently affected. So for motivation, we looked to see at how quickly after they get a correct answer do they run to get their reward, the little grain pellet that drops down. If you change the pellet reward to be a sugar pellet, they run much faster because the reward is much more salient, it's much more desirous. And what we see is that even when we have this reward system activated, they are no more interested in their pellet. And in fact, I've measured how much food they eat afterwards, and it doesn't seem to affect their hunger or how much they're interested in eating. They're pretty interested in eating already. Is this a motor control effect? Is it maybe that if we activate these circuits, they just coordinate their behavior better? And to look at that, I looked at a correct response latency, which is how quickly it takes for them to hit the window when they get it right. And there was no difference, so we're not impacting their fine motor control. And mobility, we can measure how often they go back and forth in this chamber during that 3-hour period to see whether they're more hyperactive. And aside from one animal who was a marathon runner, we didn't really see a significant effect of activating these dopamine circuits. So we can probably conclude that these improved attentional components really do suggest that we can facilitate that cognitive recovery process following dexmedetomidine. It's not exclusively possibly due to motivation and other related factors. Now, of course, we had to test ketamine. And with ketamine, even after the animal's right, if you activate that dopamine circuit, you have no impact on their cognitive recovery. The difficulty animals experience performing an attention task under ketamine, that dissociated state, is not improved, much like the rest of our experiments stimulating with ketamine. It's also not impacted by motivation, motor control, or mobility. And because we have this tool where we can target selective nuclei, I've also looked at noradrenergic systems in the brain following ketamine, activating the locus coeruleus to increase norepinephrine content. And not only do they not improve with that, which is locus coeruleus being important for attentional regulation, but they actually get worse. So overall, why is ketamine resistant to the arousal promoting effects of dopamine and also norepinephrine? So far, everything else we've used, we've been able to either improve the return of consciousness or cognitive recovery using drugs, deep brain stimulation, optogenetics, chemogenetics, and ketamine has been resistant to all of these. Well, I don't really have an answer right now, but we have some theories based on how ketamine works. It's somewhat different from the other general anesthetics. It has this unique feature where it actually increases these subcortical arousal structures despite the fact that it produces this unconscious state. And there were some studies where they actually depleted norepinephrine in the locus coeruleus, which would, without the norepinephrine, you'd normally think anesthetics would have a stronger effect, and actually ketamine got weaker with less norepinephrine. And there's also, theories out there where most anesthetics, possibly there's a component of this bottom-up processing where it's driven by subcortical structures that can no longer communicate to modulate cortical connectivity, or like thalamocortical connectivity, whereas ketamine may be more regulated by cortical connectivity, and disrupting that, you can't override that with subcortical circuit activation. All right, so in summary, dopaminergic neurons in the ventral tegmental area as a pathway to recovery of some types of general anesthesia, well, several types of general anesthesia. We can do it pharmacologically with deep brain stimulation or with circuit-specific activation in order to restore behavioral signs of arousal and neurophysiological signs of arousal, but also we can use the circuit-specific activation to also improve cognitive or behavioral signs of cognitive function. And with that, I would just like to thank my mentor, Ken Stoltmeier, and I work with a lot of people who have been involved in training these animals to do these games, and our funding sources. And thank you very much. Happy to take any questions.
That's beautiful work.
Thank you.
And I love the way you're able to tease apart the different components like motivation, et cetera. Did you ever try the opposite experiment where you take the dopamine neurons down, like use halorhodopsin instead of channelrhodopsin?
I have not done it with the inhibitory optogenetics or inhibitory dreads. I've What I've done, though, is I've used a dopamine receptor antagonist systemically, and it essentially makes recovery impossible following isoflurane, sevoflurane, and propofol. They stop doing the task. So whether that's because they're sleepier or now they have no motivation, that's hard to do because they're really uninterested in performing the task. But yeah, it's much easier to make animals worse at an attention task than it is to make them better.
Yeah, no, that's a good way to do it. Thank you.
Thank you.
Take the next microphone.
So great presentation. I'm not sure if you were here for the quantum biology part, but there was an explanation that anesthetics appear to work by disrupting the brain's ability to do quantum processes. So what I'm wondering is what you think about that, and if you think that sort of the performance of these rats improving on the cognitive tasks when you use this chemogenetic intervention, while they're still under anesthetic, has any impact on the kind of ability of the brain to do quantum processing. So the anesthetic is still there. Presumably they can't do quantum processing. So if your model is analogous to a human being, like if you could do this experiment in humans and they reported being conscious when you did the chemogenetic intervention, would you say that this falsifies the notion that anesthetics work by disrupting quantum processes?
That's a really great question, and I didn't see that panel. I was looking at the sleep and dreams panel, but no, I'm vaguely aware of the quantum explanation for general anesthetics, and I'm not going to lie, it goes far over my head and in how that exerts effects. I think it would be a good model to attest if you can capture that aspect and you can see the difference between activating these circuits. Now in humans, I think there have been a few clinical trials that have started to actually do dreads in humans for some disorders. It's very preliminary, and I don't really think this is somewhere I'm going to go with the research. I would not recommend injecting these viruses into people, but that's a possible direction for people to take it in. But that is really far outside of my field. But great question.
Thank you.
Thanks. This relates somewhat to the first question. I was thinking this must have some direct clinical application. So I would have thought that that anaesthetists or others would be using quite a lot of dopamine antagonists in the immediate post-operative period to prevent people vomiting. And I happen to be a clinical neurologist, and I'm always asked, what are we going to do about my Parkinsonian patient and their dopaminergic therapy perioperatively? And I tend to say, oh, well, just give them a rotigotine patch. It won't be as good, but they'll get a bit. But maybe I should take it more seriously and really make sure that they get sufficient dopamine so that they'll wake up after their anesthetic.
So that's a really good point. So the interesting thing with, and you might have a better answer for this, but it's interesting that like L-DOPA doesn't seem to be a treatment for ADHD, and when we implanted we did implant these stimulating electrodes into the substantia nigra. It doesn't do anything to the animals. It's very peculiar. So it's not all dopamine everywhere. It seems to be a pathway selective dopamine effect. So I'm not too sure why L-DOPA seems to work for Parkinsonian disorders, but it doesn't work for ADHD, whereas as far as I know, you don't treat Parkinson's with methylphenidate or Adderall, right? And yet both of these should increase dopamine. So I don't understand why these have different effects on ADHD and Parkinson's, but it may be one preferentially increases activity in this mesolimbic, mesocortical pathway, whereas the other has a more profound effect in that nigrostriatal pathway. Either way, I wouldn't make recommendations on how to treat your Parkinson's patients after general anesthesia, but that's a really great question.
Thank you.
Thank you.
So you're working on various switches of controlling consciousness in the brain stem. I was wondering what your perspective was on the older hypothesis that has to do with the ascending RAS system.
Descending reticular activating
Yeah.
system in what context?
In the context of brainstem control mechanisms of consciousness.
So it's true. We don't know if the activity we're doing, when we do directed activation, we only do the upstream activity. We're only targeting these arousal circuits to higher order circuits, like the nucleus accumbens, like the cortex. So I don't know if activating these descending pathways would have the same effect. Usually with anesthesia, there's a lot of spinal inhibition already on board. So clinically, whether it would make a difference, I'm not too sure. But I don't know how it would affect the downstream effects, because we also don't have a great way of.
I'm talking about the ascending RAS.
Oh, okay. Sorry. I don't know.
I mean, the point is, the whole concept seems to have been lost. I was wondering if you knew the relationship between what you were doing and that older concept.
Sorry,
No.
I'm not sure.
Hi. Thanks. It's very exciting experiments that you have done. Just wondering something about the cognitive function testing when you are saying that the clozapine N-oxide, did you say? It's an antipsychotic. Interesting that it is helpful for recovering of the cognitive functions post-anesthesia. And it's dibenzodiazepine group of molecules. So I was wondering, a lot of times in anesthesia, you give midazolam as a augmenting thing as well in the most of the procedures. So would that make any difference in terms of cognitive return?
Sorry, would clozapine N-oxide on its own?
Yeah, the clozapine N-oxide is definitely helping, but it's an antipsychotic. That's something else to wonder about. But the clozapine is a dibenzodiazepine group of molecule. But benzodiazepine group, a lot of them you give for sedation or during the anesthesia. And one of that is midazolam is commonly used. So midazolam, I don't know if it's different here, or lorazepam or midazolam or something like that. So if you are giving that, is the graph that's a difference in terms of getting back to cognitive function, would that be adjusted? Because you see a lot of them clinically not being disoriented when they are recovering.
Right, yeah. Well, I know this more, I don't do clinical work, but I know that with patients that have a higher chance of developing something called postoperative delirium or postoperative neurocognitive dysfunction, the recommendation is to not use benzodiazepines. Exactly.
Yeah.
It's shown to have a deleterious effect on the cognitive function. So right now the chemogenetic studies have really only been tested in an alpha-2 adrenergic receptor agonist drug and the NMDA receptor antagonist ketamine. So I can't speak to how it would look like with a benzo. The only reason I haven't tried other GABAergic anesthetics is young, healthy animals recover very quickly, and so you can't improve it. But I'm working on doing these tests in aging animals and animal models of Alzheimer's disease, wherein even recovery from situations like regular anesthetics or benzodiazepine, where normally it would be fine to recover in, creating models where it's more challenging for those animal systems to see whether at that point can we use this as an intervention for those kind of anesthetics. Right now, I don't know. I haven't tested the benzodiazepines.
No, because you're mentioning about the other model where you were giving amphetamines or methylphenidate, where they get awakened sooner. Wonder if some of those post-operative confusions that we have is something like they are active physically, but not yet there cognitively, and that's causing a lot of delirium kind of presentation.
Right, I think that's a very valid point. There is two different types of delirium. There's hyperactive delirium, which is the one that you're referring to.
Yeah.
And that's the one that clinicians find more problematic because they're pulling out the tubes. Hypoactive delirium is much more common, but because the patients are sedated and inattentive and delirious, they're a good patient still, so it tends to not be something people are trying to recover. So I do think that depending on what problem you're trying to solve, which is the more common hypoactive delirium, which I think maybe is better modeled by maybe the dexmedetomidine, where they're hypoactive for a long period of time. These tools might be more effective for that, for like boosting up activation and activity and restoring cognitive function, whereas someone who's already hyperactively delirious, probably hyper-arousal already going on, I probably wouldn't suggest giving them amphetamine.
No, I was thinking more in terms of midazolam being routinely used, and it can actually help for even recovery of cognitive functions, and it's less harmful than a broad spectrum dibenzodiazepine like clozapine, because again, it's on an NMDA receptor antagonist GABAergic activity, so learning and they're getting back to their task, what you're measuring there might be impacted with that pathway as well. But in terms of answering that ADHD treatment not working, I think the issue is that of the firing tone of dopaminergic neurons and the balance between other neuromodulation of locus coeruleus as well as the serotonergic thing. The serotonergic part was distinctly absent in some of that. I think it's sort of a permissive action for development of that conscious experience, isn't it?
Yeah, there is working on the serotonergic system as a, I think Dinesh Pal is working on like psychedelics right now as a potential reversal mechanism. So that's some more recent, that's very cutting edge research that is going on there that I'm aware of. Yeah, dopamine is not the entire story, absolutely. And the maps that I show, there's the dorsal raphe nucleus, stimulating that with the serotonergic activation certainly it facilitates arousal. It hasn't been done in the context of any kind of cognitive recovery. Most of these studies kind of cut it off at the point where the animal flips over. We call the day over and we stop looking at it. But I think this is kind of helpful to include this cognitive component because exactly we don't really know too much after we've activated these systems what's going on in their brains.
Because even in the yes, sir.
Hi, great talk. I was wondering about the mechanism. I am no expert in anesthesia, but what's so special about the reward pathway that others don't have?
I think that's a great question. Well, originally there was some work that used these transgenic mouse models. They were called dopamine deficient mice, and they weren't able to produce dopamine, and pretty quickly they die off because they're not motivated enough to feed themselves, do sort of basic functions. And there was some work also in the sleep literature showing that, activating this reward pathway, we call it a reward pathway, it's involved in a lot of functions. It's just the one that I think that gets the most widespread communication because of addiction. But if you are stimulated enough, you will delay going to sleep, right? If you're interested, you'd rather play more of an interesting video game or read more of a book than falling asleep. And so it does seem to be this interconnection between, this motivation, reward, whatever you want to call it. That's a great concept for describing a circuit that's probably much more complex than that. But it does seem to be this arousal promotion, right? If you are trying to trigger reward, you at least need to be aware and cognizant and alert to that reward.
Thanks.
Hi, amazing talk. I don't know if this was just coincidence, but I noticed that you referred to a lot of the animals as she, and I was just wondering if you only tested in female animals, as I know there are sex differences in the effects of, especially drugs for ADHD.
Thank you, great question. Yeah, I do gender my animals when I refer to them just because I can tell that's a male versus a female. I do look at both. We always do sex differences. And specifically for dexmedetomidine, we saw very robust sex differences. And not only that, saw differences in the estrus cycle too. So even within some animals, you see the difference in the duration of anesthesia with changing in their estrus cycle, which is analogous, I guess, to the menstrual cycle. It's very different. But with the stimulation, we don't observe sex differences. And it could be because the stimulation is so strong, like with e-stim, opto, and even chemogenetics, and of course with the drugs. The effect is so strong that you can't distinguish between males or females, even though that the females take much longer to recover from some of these drugs. So it's hard for us with these paradigms to distinguish whether our treatment has differences between the sexes. But absolutely, our anesthetics we test, they do have different impacts, but we can reverse them all with a sufficiently strong stimulant. Great question, thank you.
Hi, thank you for that great talk.
Thank you.
Is the implication that VTA dopaminergic inhibition is some sort of key part of a common pathway of anesthetic action, and that these techniques reverse that? Or is it more that kind of dopaminergic stimulation is sufficient to independently overcome the downstream effects by some alternative pathway?
So is the dopamine stimulation that works to restore consciousness the same thing that's restoring is that the question?
So is it sort of directly reversing part of the anesthetic pathway, or is it doing something else that overcomes it?
That's a great question, and that means I probably can't answer it. But so what I've noticed with these different paradigms we've been testing is that restoring consciousness seems to be very dependent on the frequency with which we stimulate these arousal pathways. So with our optogenetics and our deep brain electrostimulation, we can dictate the rate at which these subcortical structures are firing, and that actually has an impact on how effective it is at restoring consciousness. With the cognitive recovery, with DREADS, we don't get to choose the stimulation pattern, it's just more active. In fact, that could be why that stimulating the locus coeruleus chemogenetically has no impact, because LC firing patterns is very dependent on what it produces, with phasic versus tonic firing showing very different cognitive outcomes. So I think that we're probably looking, when we're looking at these dopamine pathways, the ones that are targeting return of consciousness, maybe activity dependent, maybe structure dependent. So if I were to activate the VTA to the accumbens versus the VTA to the prefrontal cortex, I might have a better effect at recovering cognitive function than with just recovering consciousness. But right now I haven't distinguished between those pathways with these models, and I can't with the chemogenetics right now. That's a really great question. Thank you.
We're moving on to our second last speaker, coming from Michigan. He's our youngest member, grad student in the department, not affiliated with Tony, but same school. So take it away.
Can I please? Hello, everyone. It's a nice view up here. Thanks for joining my presentation. My name is HYUNWOO JANG from Center for Consciousness Science, located in the University of Michigan. I'm a PhD candidate advised by Professor Zirui Huang and Anthony HUDETZ. Zirui will give a plenary lecture on Saturday. It's my personal honor to be here since I attended this conference as a student 9 years ago, and now I'm participating as a speaker. So I'm very glad to be a part of this academic society, and thanks for giving me this opportunity. Today I would like to present you our recent findings about the changes in balance between integration and segregation during anesthesia. I just want to give you some of my backgrounds. I was born and raised in Korea. I translated 5 books about consciousness. 2 notable books are Conversations on Consciousness, excellent book written by Susan Blackmore, Husitusen, and Universe of Consciousness by Edelman Tononi. By the way, to give you a short Korean language lesson, in Korean language, the word for being awake or conscious is called 깨어나다. 깨어 means to break out of something, and nada means out or being alive. So it literally means break out of something. I think it's linguistically conveys some emphasis on the dynamic aspect of consciousness, which may be related to Eastern way of thinking about our mind as opposed to Western culture, but I'm not sure. But then what are we breaking out of? So to study this question, I joined the group of Professors Ree and Tony using anesthesia as a tool to modulate consciousness and see how our brain dynamics change when we lose and regain consciousness, which is the topic of this workshop. So first I will introduce you the concept of integration-segregation balance and a novel metric that can measure the balance. And then we will apply the metric to an anesthesia data set to reveal what happens during loss and recovery of consciousness. Yeah, and I'll give you some speculation if I have enough time. So the key concept I want to deliver to you today is the integration-segregation balance. In the cognitive neuroscience, the main tenet is that there are certain brain areas that do specific tasks. A famous example is the Brodmann area, which is the distinction of the brain area based on the cytoarchitecture and still valid today. We know that brain works in a parallel manner, and there are a lot of unconscious local information processing going on. In more psychological manner, our experience is also comprised of many components, such as 5 senses, thoughts, action planning, or so. So we can say the brain areas and our experience itself are, in some sense, modularized, which correspond to the concept of segregation. But this is not the whole story. We don't experience these components individually. Our brain somehow unifies these modalities into one unified, coherent experience. So how this is possible, it is also referred to as the binding problem, as you guys may have heard of. Related to this, Bernard Barr suggested global workspace, suggesting that there will be a system or a mechanism that integrates distinct parts so that the brain can make a coherent information processing. So this mode of action corresponds to integration. So now we can see the conscious brain has to achieve 2 seemingly contradicting tasks, which is segregation and integration. If the brain is too much integrated or hypersynchronized, it'll, in an extreme case, it'll correspond to seizure. If the brain is too much segregated, the unified experience will not be formed, such as anesthesia and a disorder of consciousness. And studies report that the brain is actually alternating between 2 modes. And it is known that during the altered state of consciousness, like anesthesia and psychedelic states, this balance is disrupted. Especially during a psychedelic, we have a preliminary finding that the LSD relates to hyper-integrated stage, and there is also a reference for that. So since we are in the loss and recovery workshop, I'd like to ask a very intriguing question. I always like to think anesthesia as a rebooting our brain as an analogy. So when we hit the restart button in the good old Windows XP desktop, it shuts down and reboots. And so have you ever wondered during the process what happens behind the scene? There will be a boot sequence, that is, there will be a temporal sequence of components of the computer that will turn off and on first. Especially in the context of integration-segregation balance, we may ask which brain part or subnetwork changes first or later? I will get back to this question at the later part of my presentation. So I introduced the concept of balance, but it so far is a conceptual thing. Therefore, there is a gap of knowledge about how to measure this balance. To understand this, I would like to introduce a history of this concept and how it evolved. So this concept of balance was first proposed in 1994 by Giulio Tononi et al. At first, it was the restatement of the binding problem that I explained earlier. And Tononi proposed the concept of neurocomplexity, which I will not go into detail, but Tononi argued that this can measure the segregation integration balance. So this metric is based on information theory. So if you select certain part of the certain elements from the network and calculate the mutual information, Tononi argued that the deviation of that mutual information from the linear trend will correspond to both functional segregation and integration. Then it will correspond to the balance of the system. However, this metric is a little bit computationally infeasible, and its usefulness is a little bit in question. By the way, anyway, partly inspired by this, Tononi eventually formulated phi that calculates the integrated information or the size of repertoire of a given causal system. For details of IIT, there will be so many great lectures in this conference I'm also waiting for. So anyway, if I emphasize a little bit, this shows that the integration-segregation balance is an important theme that runs through the whole core of consciousness science. It is because the balance is the attribute of our consciousness itself and also the neural system that enables consciousness. And this balance has been widely studied in the perspective of network neuroscience. So network neuroscience is a branch of neuroscience that converts the brain signal into a network, just like our social network, and analyze the network properties. So what is network? Network is a type of graph. Graph is a set of connections with of a given elements or nodes, and network is a graph with an attribute to the connections, such as weight or the connection strength. So not all connections are equal. There are weak or strong connections which actually correspond to the actual brain network. So in the context of network science, visually, we can easily imagine a segregated or integrated network, just like the figure shown here. For our consciousness, as I said, both extremes are not favored. We have to be in a right amount of integration and segregation. So how to measure the network balance? Before I go into more detail, let me briefly explain how we can construct a network from a brain signal, especially fMRI. So what fMRI measures is the ratio of deoxyhemoglobin and oxyhemoglobin in the capillary at a certain brain area. We know that the amount of oxygenated blood is correlated with brain activity happened several seconds ago. And this is called neurovascular coupling. And we can take this phenomena to estimate the brain activity. So basically, we scan the brain with an MR scanner and extract the time course of the signal called BOLD signal, blood oxygen level dependent signal, from the regions of interest. Then this serves as a proxy for the brain activity of a given region. And what we can do to estimate the interaction strength between region A and B is to calculate the Pearson correlation of the BOLD signal. And Carl Friston defined it as a functional connectivity of region A and B. So if we repeat this process for every pairs of regions, we can get a functional connectivity matrix, and then we can reconstruct the network by taking the connectivity as the correlation values as a weight or connection strength. And if we calculate the correlation within the entire experimental plan, time scan, it'll be static. But in fact, the brain network keeps reorganizing in a dynamic way. So the trick is to capture the dynamics in a varying time window and see the dynamics of the brain connectivity pattern. So it's called dynamic functional connectivity. So now that we have a series of brain networks, we can now measure the integration and segregation. So in the network science, segregation is often measured by the clustering coefficient. The clustering coefficient of a given node is defined by the connections between the neighbors divided by the number of all possible connections between neighbors. So a very simple example, the node 1 here has 3 neighbors, but the neighbors are not connected to each other. So in this case, the clustering coefficient of node 1 is defined as 0. If there is one more connection between 3 and 4, then the clustering coefficient of node 1 becomes 1 over 3. In the case of fully connected, the network, the clustering coefficient becomes 1, meaning that the community around node 1 is highly segregated. So in short, if your friends are friends themselves, it means your community is highly segregated. So in a more global level, you can obtain the global clustering coefficient by calculating the average of the local clustering coefficient. So you can see in the network in the left, which is actually a randomly connected network, shows a low clustering. There are no well-defined segregated clusters, which results in the low global clustering. In contrast, the network on the right shows well-defined clusters, meaning that the network is well segregated. So we can use the clustering coefficient as a proxy for segregation. And the integration, on the other hand, can be defined by the degree of which a given information can be easily flowing or be communicated with within the given network. So let's say you have heard a surprising gossip or news, and if your community is highly integrated, then that gossip will be readily shared by all of your community within a few days. But if you are rather isolated or doesn't communicate with your colleagues that much, it will take much longer time for the message to be spread. So if the information in a node 1 wants to be delivered to node 7, it takes 3 steps, at least 3 steps, which is the shortest path length. So we can take the inverse of the shortest path length and define it as an efficiency between node A and B. And then we can define the global efficiency by taking the average of between all possible pairs of nodes. So the network in the left, which is called the lattice network, is an example of network that shows a low efficiency. And however, the random network shown on the right shows high efficiency because there are a lot of shortcuts you can traverse if you want to go to the other side of the network. So therefore, the efficiency can be used as a proxy for integration. So as you can see in this example, achieving both high integration and high segregation is not a trivial task. It needs a specific network structure. So what we need is to carefully define a metric that can measure the balance. There are a lot of metrics that measures integration or segregation in isolation, but there are not so many metrics that measures the balance. So the metric that I showed is well defined in the binary network, but actually there are weak or strong connections, as I told you. And What I did to address this issue is to binarize the network with a varying threshold and calculate the efficiency and the clustering thereof. And then we integrate the efficiency and clustering over a threshold to obtain the multi-level efficiency or clustering, which we used as an index for integration and segregation. And then we define the integration and segregation difference, or ISD, as the subtraction between two. So if a network is at a balance, the ISD should be zero, near zero. And a negative ISD means it's predominantly segregated, and positive means integrated network. So let's apply this metric to real data and see what happens. So the first thing we did to test this metric is to apply this on the large human connectome project data set comprised of 1,000 healthy awake participants. The value of ISD was around minus 0.05, suggesting that healthy awake brain is at the balance or a slightly segregated state. However, there are some individual variability as well. So now that we confirmed the possibility of ISD, we applied this to our 2 anesthesia data sets, which includes total 45 subjects. The anesthesia was conducted with propofol infusion, and the old data set in 2 data sets includes both baseline and loss of responsiveness period and recovery period. And importantly, our data set also includes the behavioral transition period as well. So this enables us to see what happens during the process of being anesthetized. So with the 2 data set, we independently observed that the time course of ISD shows a significant decrease under anesthesia. You can see that during the baseline, the mean ISD is near zero, but it decreased to around minus 0.1 during anesthesia. This suggests that under anesthesia, our brain becomes segregated. And we also verified that all predefined 8 subnetworks undergo segregation. More on the subnetwork later. And then we plotted the ISD against the propofol concentration at effect size. And we see some kind of inertia or lagging of ISD change for whole brain and all subnetworks. So this well corresponds to the concept of neuroinertia, which Tony has mentioned. And we plotted the distribution of the brain states within the 2D space of integration and segregation, and observed that during a loss of responsiveness, the brain moves towards segregation. Here, the dashed line is ISD 0, so balanced state. You can see the center of the contour line is moving to upper left, which is segregation. Of course, the ISD value shifts towards left. Let me skip this. So based on this finding, we benchmarked ISD against currently used balance metrics. So the performance of a certain metric can be evaluated by analyzing the receiver operating characteristic curve, or ROC curve. So in short, the classifier is, if the classifier is more close to the blue curve, it is better classifier. And we observed that the ISD was the best metric compared to other network metrics like modularity, participation coefficient, etc. So next thing we did is to align the ISD curve with the onset of loss or regain of consciousness, because people lose responsiveness at a different time and dose. So by time locking the ISD around the behavior transition, we can see that on average how the balance changes when we lose or regain consciousness. So during the induction, ISD starts to decrease right at the moment of right at the moment of loss of responsiveness, and during the emergence, ISD starts to increase around like 4 minutes before recovery. So this is this kind of shows that there are some asymmetry in terms of ISD recovery. And the question that I asked in the first part of the lecture, if there is a boot sequence of the brain, let me apply this metric to subnetworks and see what happens. So within our brain, there are well-known brain networks. Everybody named them differently, but in our study, we defined the following 8 networks. The first is unimodal, which means they deal with only one modality, and this includes visual and somatomotor. And there are transmodal networks such as default mode or frontoparietal. They engage in multiple modalities and higher order functions such as thought and problem solving. And also there are attention networks and limbic and subcortical network as well. So what we ask is, are there any sequence in the chain during the unconsciousness transitions? So you can see in this plot that we can see that the visual and somatomotor is disintegrating first, followed by the default mode and the frontoparietal. Lastly, the subcortical network disintegrates. So there seems to be a hierarchical order. Unimodal disintegrates first, followed by transmodal and subcortical. So during the emergence, we found that there is a common sequence of reintegration. So this time too, visual and somatomotor reintegrates first, followed by transmodal and subcortical. And when I plotted the amount of change, you can see the somatomotor and visual and attention networks reorganize a lot compared to limbic and subcortical and transmodal. And we also tried to use the integration and segregation values of subnetworks and the whole brain as features to train machine learning models to predict the conscious state, awake or not. So random forest network, model achieved around 93% of accuracy, showing the best performance. And important thing is that when we learn this, when we train this model, we didn't show the transition period. We trained only with a fully awake or fully anesthetized state. And however, the model could track the behavior responsiveness in a high accuracy of around 90%. And especially around the transition period, the prediction of awake showed steep decrease or increase. And I also investigated the relationship of balance and the metastability. So even during you guys hear my lecture, you will have a stream of thoughts. So this can be explained by the concept of metastability. So if you engage in certain thought, your brain overcomes a certain type of barrier and fall into another valley of the landscape. So metastability is defined as the appearance of the non-equilibrium state, and it measures how easily the brain can navigate the energy landscape. So practically, it is measured by the moving standard deviation of the mean coherence. So in short, if the brain keeps synchronized and desynchronized repetitively, the mean coherence will fluctuate a lot. Then we use the standard deviation as a proxy for metastability. And what we found is that under anesthesia, indeed, the metastability decreases. And what we found is that the integration-segregation balance is also highly correlated with metastability, especially between the integration and segregation. Integration was more related with metastability. Another important property of the brain signal is complexity. As Tony illustrated beautifully, complexity has been suggested as an important property that is related with consciousness. So the conscious brain will create a non-trivial, meaningful signal, which corresponds to the concept of complexity. So there are no globally accepted metric to quantify the fMRI data complexity. So to address this gap, I implemented a complexity measure called pattern complexity. So we converted the bold signal into the pattern sequence, and we combined 3 metrics that measures the certain aspect of the complexity of this sequence, and then we combine them into to create a pattern complexity. So we found that the complexity indeed decreased during anesthesia, corresponding in line with the literature. And also ISD and the complexity was generally highly correlated. Also complexity was this time tightly correlated with segregation compared to integration. And we replicated this finding in the sleep data as well. And to give you a summary, we devised a metric that can measure integration-segregation balance based on network neuroscience. And then we investigated the booting sequence or rebooting sequence of our brain, showing unimodal changing first, followed by transmodal and subcortical. And we used machine learning model to try to use integration segregation as a predictor for our conscious states. And lastly, we found that this balance is also differentially related with beta stability and complexity. I think I have 2 minutes left, so let me show you like another view of integration segregation balance that I recently learned from the papers of Andrea Lupi and Emmanuel Stamatakis. So when we say integration, not all integration is the same. So there are type of information, so you can decompose you can decompose the information in 3 different ways. So if you see this diagram on the left, there are unique information like nuts or bananas, and there are shared information like redundant information like the square, and there is a synergistic information of the depth that can appear because two eyes work synergistically. So in that way, the information can be partially decomposed, and so if we combine this concept, this is a very nice figure in a paper of Lupi et al. I really like this diagram. It shows the whole picture. So unimodal areas engage in more redundant information processing, while synergy is majorly mediated by the transmodal regions. So my current interest is to how to merge this way of thinking with integration, segregation balance. So redundancy is some kind of integration within the module, or segregation would be, in a global level, the highly integrated subnetwork will be maybe corresponding to segregation. So it's this integration segregation balance is seems to manifest in a hierarchical manner. So I'm not still sure how to measure this like multi-level hierarchical order level balance, but I believe combining this way of thinking will reveal us the full future or full picture of information processing happening in our brain. Yeah, thank you so much for hearing my talk. Yeah.
Thank you. That was fascinating.
Thank you.
I am struggling a bit with your ISD metric, integration, segregation difference, because if I understand it correctly, you could have an ISD of 0 but have very little segregation or very little integration. So wouldn't it be better to look at something like an ISP, an integration segregation product, where to have balance, but to have lots of both would be ideal?
That's a great question. That was the question that I also encountered when I was developing this. And one example I didn't show in this slide, but when we actually increase the dose, we can see both integration and segregation decreases. Still, ISD is negative, but you can see both integration and segregation decreases. So I believe there is there will be a certain aspect of the neural system that can be captured by the ISP you have proposed. But ISD captures certain aspect of the balance, and the product actually may correspond to the small wordness, or which is the important concept in the network neuroscience, and there are some literatures about that. Yeah, thank you.
Thank you for your talk. Very exciting work. My question is in regards to the interindividual differences of the integration-segregation balance. I guess the question is in two parts. First, what would you speculate integration-segregation balance would look like in a clinical population? And then further, I guess more broadly, how would integration-segregation balance track with phenomenology? What would an integrated brain feel like as opposed to a segregated brain within a normative degree?
That's a great question. So first, as neuropsychiatric disorders, we have initially tried to calculate the ISD in a whole brain level, but we didn't find a statistically meaningful change. So I think still the neuropsychiatric patients are still conscious. So I think there is a subtle difference, maybe in a regional network level or local, like a nodal level change. So I think we need a different way of approaching that. And for the second question, what was that?
I guess in a normative neurotypical population, how would it track to phenomenology?
Yeah. That's always my interest as well. But so the dynamic window that I used is around from 30 seconds to 8 minutes. So the data I showed is 4 minutes. So it's kind of hard to imagine how our experience like fluctuates in a like 4-minute window. It will be vaguely correlated with our like vigilance or attention level, but I am not quite sure about that. But the experiment we are actually dealing with is to modulate certain deep brain areas like thalamus to see the behavioral or perception change. So that may link the changes in the integration-segregation balance mediated by deep brain areas to our conscious perception or phenomenological level. Yeah.
Thank you.
Thank you.
I thank you for your talk. On your boot sequence, it seemed like you had the 8 functional areas and how they went into anesthesia and came out of it. And one thing is, I wasn't sure how significant that was in your measurement. If you feel like it's very strongly in that order, and also it looks like a lot of the areas went induced and emerged in the same order, which is a little bit different, I think, than how a computer works. And especially having been under anesthesia, it made sense that maybe the visual would stop first. But I was surprised that the visual would come back. And does that mean, did any of that surprise you, and does that have any meaning?
Yeah, so first of all, in terms of the significance, I didn't mention much about how I calculated this. So from the ISD curve, there are 45 participants. So I can get a smoothed curve and then get a derivative or slope. Then I will get a slope distribution of 45 participants. And then I test against 0. So see if the collective behavior of ISD is shifting towards negative or positive. So that's how I get the temporal regime of the change. So I think it's statistically meaningful. And second, for the emergence, I was also surprised by to see this as well. There are some, like, if I control the parameters in a, like, how should I say? At first, we observed the unimodal and transmodal didn't like just see very much difference, but upon tuning some smoothing parameters, we could get this order. But one thing I can tell you is that the emergence process, there are more individual variability, and I don't want to like strongly argue that the order presented here is like canonical or applying to every loss of consciousness. It's specific to our data set and a propofol data set. So yeah, it would be very helpful if I could apply this to like transition in a sleep data or other kinds of unconsciousness.
Thank you.
Thank you.
Hi, wonderful talk. Forgive me if I missed this in the talk, but have you done any kind of ground truth testing of the metric? So for example, in those early Tononi papers, he showed that if a system goes from randomness to order, complexity will increase, peak, and then decline again. Would you expect your measure to follow or track that type of change? And again, have you assessed this with known signals, like simulated signals?
So first of all, the complexity measure that decreases upon random signal. I also try to like find the appropriate measure that follows that trend, but there are not so many that are applicable to fMRI. So I think my metric will be maximized in a random signal. That's what I guess. But there are some approaches to like tweak the definition of complexity to make it as a inverse U-shaped. But for this specific metric, that I didn't use that. So that's a future approach as well. And the second question was.
Well, did you just do any ground truth testing with known signals?
Yeah. So I didn't show here, but I tried using the simulated Ball signal from the known structural connectivity connectome data set. So I used the Kuramoto model to simulate the functional connectivity, and I saw that the ISD becomes 0 at certain level of noise and the connectivity at like a intermediate coupling strengths K. So I'm not sure it suffices to your question, but we tried to verify that this metric indeed becomes 0 or near 0 at a reasonable range of the parameters. Yeah.
Great. Thank you again.
Okay.
Great work.
Thank you.
Well, last but certainly not least, Stuart's going to come up and tell us that you don't have to worry about neurons and stuff at all. Just focus on the microstructure inside the cells.
Thank you, Bruce. I couldn't quite hear you, but I appreciate it. Okay. All righty, thank you. Glad to see everybody here. My topic is microtubules are the sites of anesthetic action on consciousness. And the picture is the famous first public demonstration of anesthesia at Mass General in 1846, I think. I've been doing anesthesia a long time. I'm not quite in this picture, but didn't miss it by that much. Soon after that, actually the same year, Claude Bernard did the first study of anesthetic mechanisms. He showed cytoplasmic streaming in an amoeba, a slime mold amoeba, was reversibly inhibited by chloroform anesthetic gas. Exposure just stopped the flow, the amoeboid movement, and when he got rid of it, blew it off, replaced it with air, the amoeba resumed its activities. And in the 30s, this was shown to be by direct effect on the cytoplasm rather than the membrane. And the slime mold more recently has been shown to be able to solve the traveling salesman problem if you put it in a proper well and have food and aversive conditions here and there. So, the movement, the orientation, the direction is selected by the microtubules in the cytoplasm, and then the actin flows along with it. Much later, in 1968, Allison and Nunn, John Nunn, shown here, who I met a couple times, he was a friend of my first chairman, Bernal Brown, had a paper in Lancet, Effects of General Anesthetics on Microtubules, a possible mechanism of anesthesia. And they studied this actinospherium, which had multiple axonemes, which are, you can see in the bottom, are double spiral arrays of microtubules shown in cross-section. And when they exposed them to halothane, they depolymerized and the axonemes just disappeared. They just disassembled. However, this was at 5 Mac, 5 times the anesthetic required for loss of consciousness, but it did show an effect on anesthesia. And actually, Bernal, who was trying to get me to go into anesthesia because he was desperate for residence, knew I was interested in microtubules and handed me this paper and said, this is what you should study. And I agreed with him. So that was 50-some years ago. Much later, Nick Franks and Bill Lieb, there were studies of anesthetics in the lipid membranes because of the Meyer-Overton lipid solubility and so forth. Then membrane proteins came into play, channel receptors and channels and so forth. Franks and Lieb show that the anesthetics acted directly on the proteins, not on the lipids around them, in hydrophobic pockets, lipid-like pockets inside proteins, luciferase, which didn't have any membrane, and the emission of light was, in the presence of ATP, was inhibited by different anesthetics, roughly proportional to their potency. So this turned the attention to direct effects on proteins, and the vast majority of everybody pretty much went to look at membrane proteins, including ligand-gated ion channels, receptor proteins, as the presumed sites of anesthetic actions. And anesthetics do, in fact, bind to these membrane receptors, for example, for serotonin, glycine, acetylcholine, and GABA-A, and others. But experiments produced conflicting results. Some anesthetics opened a channel, others closed it. Not all anesthetics bind to any one receptor. So there was no possibility of a unitary mechanism as suggested by Meyer-Overton and additivity of MAC based on membrane proteins. And then more trouble arose, for example, in this editorial in Anesthesiology in 1999. Evers and Steinbach said, summarized some articles, said volatile anesthetics both enhance and inhibit excitatory acetylcholine receptors, and some enhance inhibitory receptors at low doses, but inhibitory at high doses. And so it's somewhat of a confusing picture, to say the least. They call it double-edged swords. It's kind of a pun. A number of articles in the early 2000s came out against the acetylcholine receptor, the 5-HT, opioid, adrenergic, potassium channels, and others as mediating immobility, which is when you can't say for sure the animal's conscious. GABA receptors were discredited in various ways. And finally, in 2008, Ted Eager, who was the leader of the of anesthetic mechanism research, and certainly in the United States and probably the world, kind of gave up, as Bob Pierce said, on membrane receptors, and published this paper along with some other notables saying, is a new paradigm needed to explain how inhaled anesthetics produced immobility or loss of consciousness? And 2 decades of focused investigation have not identified a channel that alone is sufficient to mediate immobility, nor combination of actions of channels seems sufficient. So he called for a new paradigm. I thought, oh great, microtubules. But instead, everybody went back to lipids, and then they returned to GABA and so forth, and this didn't seem to have much impact, although it certainly convinced me, although I didn't need convincing by then. But fortunately, people like the Eckenhoff lab started to look at kind of an overall systemic approach to the problem, and for example, using radio-labeled halothane, they found 70 proteins in brain neurons found to bind anesthetics, half in membrane and half in cytoplasm, including tubulin, the component protein of microtubules. And they then did other studies. For example, the journal Proteomics, exposure of primary cortical neurons to halothane and isoflurane alters the genetic expression of 7 proteins, 3 proteins common to both, tubulin, and 2 other proteins that don't seem to have anything to do with signaling. And so proteomics and genomics point to microtubules as a site of action of anesthetics. They also did the same group did an optogenetic study where they developed a fluorescent anthracene anesthetic that was only active when it was hit with UV light, and the fluorescent product was anesthetic. They gave it to tadpoles who have conveniently transparent heads, so they could illuminate, they could give them the drug and then let them swim around, hit them with the UV light, and they would literally go belly up. They'd become immobile. And when they sacrificed their brains for science, they found that the anthracene was bound to microtubules. And Eckenhoff said this was consistent with quantum mobility theory, which was kind of paraphrasing the ORC-OR theory, referring to anesthetic action on quantum effects in microtubules. So optogenetics also, at least this study, points to microtubules. And they also did a study, a retrospective study, a clinical study on patients on Taxol or taxane, paclitaxel, an anti-cancer drug, which stabilizes microtubules and makes them so stable they can't disassemble to form mitotic spindles. And that's how they work in cancer. But by stabilizing the microtubules, they also increase the requirement for anesthetics. So they made the patient resistant to anesthesia, requiring more. And so pharmacokinetics, at least in this case, points to microtubules as the site of action of anesthesia. So what do anesthetics do to microtubules to block consciousness? I spoke about that this morning, but basically they bind by very weak quantum van der Waals forces. And for example, to aromatic rings, which if you have 2 aromatic rings at the van der Waals radius, they'll attract each other and then will oscillate in terahertz back and forth. And it looks from a number of studies that anesthetics act to disperse this dipole and block this oscillation at terahertz. So 10 to the 12th hertz, pretty fast. How does that relate to microtubules? Well, here's a tubulin, and it has 86 aromatic rings, like I just showed you, in the tryptophan, phenylalanine, and tyrosine aromatic rings. 86 is a lot for a single protein, I'm told. And in the red spheres are where anesthetics bind, and Travis Craddock simulated this. So the anesthetics are in the middle of all these aromatic rings. And in our theory with Roger Penrose on the theory of consciousness, we thought this could be a net dipole through the tubulin connecting to other tubulins, a quantum channel through tubulin, which would connect to other tubulins and form a topological qubit that wrapped around the microtubule and could be mesoscopic and would oscillate back and forth, consistent with Herbert Froehlich's idea of Froehlich coherence due to giant dipole oscillation. So this essentially creates a mesoscopic quantum state. Anirban Bandyopadhyay studied microtubules. He's in the back here and did this set of studies over 10 years looking at microtubules in 3 different scales. can you see? Yeah, you can see my cursor. So at the highest scale, we have a network of a few neurons with several probes, and then we go smaller into a single microtubule, and then we go to rows of tubulins, and we do the same. They did the same thing by applying. If you apply a direct current, a voltage to a microtubule, it's an insulator. There'll be no current flow. However, if you sweep the frequency and you will find that at certain frequencies there is conductance, and in fact very high conductance, ballistic conductance, perhaps quantum if you could ignore the connection between the probe and the tubulin, and the conductance repeated every 3 orders of magnitude. If we start at the upper left in hertz, and then in kilohertz, then megahertz, and then going down to the next one, also kilohertz, megahertz, and gigahertz, and then megahertz, gigahertz, and terahertz. And in each case, the pattern of conductance was a triplet of triplets. So 3 peaks of conductance, and each peak had 3 peaks within it. So this triplet of triplets is showing up at multiple levels in microtubules and in some other places that I'll be talking about later in the week. So what does that mean, triplets of triplets? It shows that microtubules can be highly conductive and maybe quantum, at least at these specific frequencies. So this gave rise to the idea of a scale invariant hierarchy. Anirban has been working on what he calls the great chain, a resonance chain for the entire universe, and with triplets involved. But in the brain, it can be looked at in this scale invariant hierarchy. Now, some people talk about a hierarchy going from neuron up into networks and networks in hertz, but this takes it inside the neuron, starting from the pyramidal cell on the far left in hertz, and then networks of microtubules at a kilohertz, and then megahertz, and for individual microtubules, gigahertz at rows of these dipoles, terahertz at microtubule, and then petahertz or hundreds of terahertz, where the dipoles oscillate and where the anesthetic seems to act. And then faster at the nuclear level, and then all the way down, according to Roger Penrose's idea, to the fundamental level of space-time geometry. But between the brain, within the brain, inside neurons, we're talking about 12 orders, at least 12 orders of magnitude of scale invariant hierarchy with the same processes occurring every 3 orders of magnitude. So here's a close-up of that, just to show the triplets of triplets at the bottom in the pyramidal neuron microtubule network, microtubules and rows of tubulin dipoles, tubulin, pi resonance dipoles, atomic nuclei, and then down to space-time curvatures that we won't deal with today. So Travis Craddock talked about the study this morning, and this is a computer study he did on he and his colleagues, including myself, did on a supercomputer, and basically they modeled the quantum dipole oscillations and interactions among all 86 aromatic rings in tubulin. And this gave rise to and then with 9 anesthetics, methoxyfluorane, halothane, fluoroxine, isoflurane, enflurane, SIVO, diethyl ether, desflurane, nitrous, and then 2 non-anesthetics, F6 and TFMB, which I'll talk about now. These are molecules which follow Meyer-Overton, so they are at the site of action, but they don't cause anesthesia or loss of or immobility in animals. They bind in the same areas that other anesthetics do, but they don't cause loss of consciousness. And why is that? I think a good theory should explain this. So Travis found a spectrum, or we found at KT, ambient temperature, a spectrum of collective dipole oscillations with a common mode peak at 613 terahertz in the blue light region of the spectrum. And all the anesthetics and non-anesthetics had their own frequency emissions that are shown here. I mentioned that we have unfortunate acronyms for methoxyfluorine is meth, so not methamphetamine, and death is actually diethyl ether. But anyway, so I just want to explain that. And NITR is nitrous oxide, a weak anesthetic. So the presence of each of the 9 anesthetic gases abolished the 613 terahertz peak and dampen other terahertz frequencies proportional to their potency. Non-anesthetics did not abolish or dampen the peak or frequencies. Travis showed that this morning. The 613 peak disappeared. You can see negative peak due to subtraction spectra in all the anesthetics, but not in F6 or TFMB. They actually had a different effect, a different frequency, or actually increased the terahertz peak. And so we then wanted to, anesthetic action on tubulin seemed to be able to account for anesthetic potency relatively in the context of Meyer-Overton. We then looked at the TFMB and F6 and just assigned it a MAC of infinity or 1,000, just not enough gas to cause it. And this shows that it's within the Meyer-Overton solubility region, the oil-gas partition coefficient, just like all the other anesthetics. However, we then plotted it against polarizability. And F6 and TFMB were both higher than the polarizability of the anesthetics. So what we think this means is that when there's oscillations that are going on between the aromatic rings, the anesthetic shown at the bottom will block them, but the non-anesthetic shown in the middle in the blue here just goes along for the ride. It oscillates right along with the endogenous aromatic ring. So this would explain why the non-anesthetics, despite binding there and forming van der Waals forces, don't cause loss of consciousness because they're oscillating right along with the other, with the endogenous rings. They go along for the ride. Some others support, as part of the Templeton project comparing different theories of consciousness, we could not come to an agreement with IIT or any other theory because they are all at the level, at the higher level in terms of the membrane and networks, and we're primarily within in the microtubules, so we couldn't really come to a common adversarial collaboration. But Templeton gave us funding to do this study, and we showed that ultraviolet tryptophan fluorescence hitting a microtubule with ultraviolet light resulted in fluorescence that its propagation and duration, tryptophan fluorescence lifetime, was inhibited by two different types of anesthetic, etomidate and isoflurane. And we also showed that this propagation was a quantum effect because it was longer than the classical Forster effect, and also that microtubules are efficient light harvesters. So overall, we showed that anesthesia, the two different types of anesthetics, a gas and an intravenous soluble agent, inhibit quantum effects in microtubules. So if we go back to the multi-scale hierarchy, I want to raise another issue, which is where does EEG come from? This is the 100-year anniversary of EEG. It's done remarkable things clinically and in research, but we really don't know kind of what it's about in terms of how the different frequency bands relate to each other. As you read authoritative books like Busacki, this rhythm comes from this pacemaker, this one comes from another one. Gamma, he doesn't really know. And gamma still is a mystery. In fact, zero phase lag gamma synchrony, where the whole brain is collectively oscillating at 40 hertz or higher, is difficult to explain by membrane conductance, particularly with synaptic transmission, nor with ephaptic, because that kind of dies off. So gamma is still a mystery. In my paper with Roger Penrose in 2014, we suggested EEG in hertz. A regular old EEG was actually interference from faster collective oscillations in microtubules. And also this interference could give rise to slower cognitive events that we're familiar with, like 100 milliseconds and so forth. So interference patterns going from right to left can give us EEG and slower cognition. So we can actually, Anirban showed that we can measure megahertz from the scalp, and this is Hide Saigusa, who's here at the conference, and he's holding this probe, and on the screen you can see a triplet, and he takes it away and the triplet comes back. And we want to try this with anesthesia. We did do a study with a rat, but we think there might have been some problems with that. So we are going to be doing this study with Anirban's colleagues in Bangalore in India over the summer, hopefully, in patients in a hospital undergoing anesthesia and also comatose patients to see what their signals are. Let me run this again. It's just, I guess I can't. To see what their signals are. So we'll be doing it properly in humans hopefully this summer. So the triplet can be detected from the scalp with proper instrumentation. It's actually pretty simple. Oh, here it is again. Okay, so you can see the triplet, takes it away, it's gone. This is in a Faraday cage, but you can get it without a Faraday cage. And actually, HUDETZ will have an exhibit of this device starting Wednesday, I think, when the demos open at lunch in the evening, where you can try it on yourself, see if you can find your triplet, and we'll take a picture for you with your cell phone. Or they will. Some other things Anirban's group did was they looked at neural networks. They made these wafers with probes, that dielectric resonance probes that can measure megahertz and gigahertz. And when they impaled the network, you can see that they see the image of the neuron, but not outside the neuron, because that's where the megahertz and gigahertz is coming from. And they showed that on the left, you can see the microscopic image of the neuron. The ionic conductance is in kilohertz, so these are ions outside of the neuron. But then when you get to megahertz, you see the neuron, because only the neuron has the megahertz concentrated enough to be detected. And the megahertz and gigahertz in the dendrites and soma of those neurons correlated with firing patterns in the same neuron and nearby neurons differently than did the membrane potentials, and seemed to override the membrane potentials, showing that activity in the microtubules inside neurons can regulate synaptic function even more so than membrane potentials. And this was published in a very mainstream journal of neurophysiology, and he showed these different circuits determined by the filaments, the microtubules and the actin and so forth inside neurons. So it's functional, the megahertz and gigahertz is functional, controlling network formation and integration and firing. Anirban also, in a paper with Saxena, was the first author, has shown using Bell's inequality measurements in a pair of microtubules, entanglement between microtubules. And I think this is very important because I think we need entanglement for spatiotemporal binding. I appreciated what the previous speaker said, but it didn't account for temporal binding, because shape, color, motion, meaning processed in different parts of the visual cortex are processed not only in different areas, but at different times, and yet we see unitary objects at one point in time. And if the microtubules are entangled, then a lot of things become explainable and possible in terms of consciousness. So this is a big step. In fact, I see Andrew Duggins here. I've known him a long time. He's been asking for some kind of evidence of entanglement in the brain for a long time to prove our theory. And this is not in the brain, but it's between microtubules. So thank you, Anirban, for that. Okay, I want to address George's paper. George Mashur and his colleagues had a paper in anesthesiology recently, Consciousness and the Dying Brain. It was very interesting. However, they assume that near-death experiences and out-of-body experiences are not real. That is to say, they're real if you're having the experience, but not real in terms of leaving your body, and which is a logical thing, which most people most scientists anyway, would go along with, and therefore can be explained by classical theories of consciousness, like IIT and/or global neuronal workspace, which are the only two they considered in this paper. They did document gamma synchrony mediating these NDEs and OBEs in animals, and showed with some processing that the activity can be functional, that it feedback and feed forward are active, and it could be functional, which I think it is. However, even if we know where consciousness occurs, there's no clue what activity is critical. And this is a criticism I think IIT is going to have to answer. What is it that the integrated information actually is? Also, what is the origin of the gamma? What is happening, and also what is happening with megahertz under anesthesia? Kind of a separate question. But what if NDEs and out-of-body experiences actually involve consciousness becoming non-local and being outside the body? Well, the OR theory can explain this by consciousness by collapse of the wave function. I don't have time to go into all this, but can occur anywhere along the spectrum. And as you move to the right, it's faster and faster and therefore more intense. But when you get past, let's say, 10 to the 18th hertz and down towards space-time geometry, at some point you're not in you don't need mass anymore. You're just in space-time vibrations. And in principle, consciousness could exist outside the brain and body. So it's possible when this happens, this is the end-of-life brain activity from one of the studies cited in George's paper, this might be happening. Now, I'm not advocating for this. I just say that until we know for sure what consciousness is in the brain, we cannot rule it out outside of the brain. And so something out of body and even afterlife are not impossible if consciousness is a quantum effect. So let me conclude. I've got a number of conclusions here. First, genomic, proteomic, optogenetic evidence for anesthetic action on microtubules, I think, is more evidence than there is for any other site of action of anesthesia. Anesthetic inhibition in microtubules is proportional to the Meyer-Overton correlation, as Travis showed this morning. I don't think any other theory of anesthetic action can do that, and it's unitary. It explains why nonspecific quantum binding affects only consciousness, as the organized quantum states are randomized. And this is an answer to a question Christoph Koch asked me 30 years ago. I said, Christoph, we really got to figure out how anesthesia works because it's selective for consciousness. It only affects consciousness. And he said, no, it's too nonspecific. It binds to everything by very weak forces all over the brain, all over the body. It can't possibly tell us anything. And that was a good answer. And I thought about that for many years. And a couple of months ago, somehow it dawned on me that it's actually pretty simple. There's quantum stuff going on everywhere, all over the body, particularly in lipid-like regions. But only consciousness has highly organized quantum states, like a Bose-Einstein condensate. So only consciousness, only processes controlling consciousness or causing consciousness can, if they are randomized, will have an effect. Everything else is already randomized. So an anesthetic randomizing something other than being involved in consciousness has no effect because it's already randomized. But in consciousness, all these quantum dipole oscillations are highly organized or orchestrated, and so they're easily perturbed by decoherence by anesthetics. It also explains non-anesthetics because of the polarizability being too high. I just want to point out, when I first raised this years ago in the anesthetic world, people said, "Oh, the binding of anesthetics to tubulin is 1,000 times weaker than it is to like a GABA receptor." But then Nick Franks, who didn't agree with me, was kind enough to point out, however, there is about 1 million times more tubulins than membrane receptors per neuron. So that means there's about 1,000 times more anesthetic in microtubules than in all the receptors in any one neuron. And so it's logical then anesthetic can act on microtubules. The megahertz triplet patterns from scalp being tested for NCC likely emanate from microtubules, and we're going to test that in India this summer in patients. Entanglement can account for spatio-temporal binding and zero phase lag gamma synchrony. I don't think we can explain zero phase lag gamma synchrony and binding by classical means. The microtubule site of action directly relates to OR theory, which has, I'm biased, but I think this is true, the most explanatory power of other theories of consciousness. The most biological connection, which is dearly lacking in IIT. I still don't know whether it's axonal firing, synaptic transmissions, or what exactly is the information. and empirical evidence among theories of consciousness. And finally, OR and microtubules can account for non-locality in consciousness if it occurs. And I don't think we can say it can't occur until we know exactly what consciousness is in the brain. I'll end there. Thank you very much.
Step up to the mic if you have questions. I'll start off with a quick one. That's well done, Stuart. You missed one thing that I love about the OR theory is that it's the only theory that says consciousness is an entity. It's produced every time there's a collapse of the wave function, which happens 1 million times a second all over our brain. And if you think of it as an energy or a force, then you've got your energy or force that is consciousness. So I just wanted to add that.
So that's a good thing, right?
That's a good thing.
That's a good thing, right?
Yeah.
Okay, thank you. I think, the collapse of the wave function, it's a whole other dilemma, the measurement problem. Some people don't think there is collapse, that there's multiple worlds, but you kind of have to pick between multiple worlds or collapse. Yeah.
Hi, thank you. Thank you for that talk. I'm struggling to understand how much ORC you need for consciousness. I'm struggling to understand how much orchestration you need. Does a single neuron, a single microtubule, a single tubulin molecule have a significant?
Well, the collapse occurs by at time t equals h bar over e sub g. H bar is a constant. E sub g is the energy of superposition, separating something from itself, whether it's a nucleus in a tubulin atom or its equivalent space-time curvature. So there's a threshold when it occurs. So when that occurs, everything that's entangled in that superposition is what the experience is. Now, this happens randomly in the environment, in the air, in the table, in other parts of our body that's not involved with consciousness, and we call that proto-conscious. And the best metaphor I can think of is if you go to the symphony and they're tuning their instruments and everybody, you hear all these tones and sounds and noises. It's not melodious. That's noise. It's really noise. And I think there's proto-conscious noise everywhere. But then in the symphony, the band begins to play, the orchestra plays, although you don't necessarily need a conductor in our view in consciousness. And that's music. So the difference between the proto-conscious noise that's everywhere and your consciousness or anybody's consciousness is like the difference between music and noise.
So would that mean that there were gradations between proto-consciousness and the full thing?
Yes. So as you go from, if the collapse can occur at any point here, as I said, if it occurs far enough to the right, it can be non-local. And we think intensity also goes to the right. So in terms of the graphs that were shown earlier of content versus intensity or level, so in this view they're both related because it's the number of tubulins in superposition that collapse. So the more you have, the more content because the more possible permutations of the tubulins. But the intensity of the experience is related to that also, like a photon. The higher the energy, the more experience. So we can account for both content and level of intensity as was discussed earlier.
Okay, thanks. Can I ask a really small addendum to that? There's 5 people.
Thank you for your talk. There's been quite a few studies out of NYU in the recent years using hyperscanning neuroimaging, identifying brain-to-brain synchrony between cooperative groups. Particularly given the latter half of your talk, do you think it is consistent with your microtubule hypothesis that brain-to-brain synchrony could be a substrate whereby a group consciousness may be formed?
Entanglement between people?
Exactly.
I think it's possible. I think at least some of the time, telepathy, that sort of thing. Now we're seeing all kinds of reports of synchrony among EEG and so forth. I don't know whether that's quantum or not. But it's possible. If you know, it's kind of it's a Pandora's box. So if we have non-locality, many things become possible. I think that's a good thing.
Thank you.
Hello, thank you for the talk. Every time I listen to the lecture of you, I feel inclined to the microtubule theory, but soon I kind of get back to the receptor theory. So I want to ask what experiment or finding can induce a genuine paradigm shift from receptor view to your microtubule view of anesthesia?
Wake up. I think, you know, go back to Eager, 2008. Why did everybody forget about that? I mean, why? I think you're, no offense, but I think you're beating dead horses looking at receptor mechanisms.
Stuart, a really good talk. I have a question. If we start with the very right-hand side, from my point of view here, like you mentioned, consciousness can exist in that collapse without the need for microtubules. So now in a brain, in a neuronal structure, if the microtubules are the sites of the megahertz and these vibrations, would you say that that is sort of a manifestation, but it still requires biological functions downstream or upstream? Or let's say if the collapse is happening at the microtubules, still there needs to be further signal transduction, further mechanisms for the experience, the full experience of consciousness. So consciousness out in space is one, which is not being experienced by anything. But then the role of microtubules is to bring that information or collapse and translate into experience. That will still require further downstream mechanisms. I don't know if you've thought about that, or would you say that to that?
I'm not following you, Rajneesh, entirely, but when the collapse occurs, it selects new states of tubulin. So the tubulins are in one state, they go into superposition, and they're entangled, and they collapse, and they pick a new state. And that set of states are the initial conditions for the next process, and it has downstream effects. So that's how, you know, you decide you're going to do this or you do that by what tubulin states are selected. That's how you get causal action and even free will that way, or we call it free will. I think it's a combination of things, but it selects the new states which cause downstream effects.
So quickly, if you remind, I'll just add. So for example, we've done these experiments in plants, and plants have stomata which are made with guard cells. And we see this when we apply isoflurane, we see disintegration of microtubules. What is known about how stomata work? Microtubules provide a cytoskeletal structure that allows water pressure to come and open the pores. So there are other processes that rely on microtubules.
Yeah.
Right. So I think that's where I'm going. So there has to be some interaction with other processes, which then ultimately provide the function.
Yeah.
I think to maybe solidify this theory further, we need to find what other proteins or other things microtubules interact with, so that the function part, that pathway, is explored.
Yeah, well, that could be, but at least for the quantum period, they have to be isolated. Once the collapse occurs, they can interact with the outside world. I mean, it's always a problem of how you have a quantum system that's going to receive inputs from the classical world and then put outputs into the classical world. And the answer is, actually was in Roger and I got this idea from an AI, a story by Paul Benioff, one of the inventors of quantum computing, about a quantum robot that alternated, had cyclical phases, and of quantum classical, and I think that's exactly right. So each collapse is quantum, it's classical interacts. I just want to add that Rajneesh has done work on plants with phototropism and things like that are inhibited by anesthetics. And you think it's the microtubules, or you know it's the microtubules?
We know it's the microtubules. It is pretty clear. In fact, we're looking at, because when plants, we can look at mutants, which are lacking certain aspects. That's very hard to do with animals and humans. So we are pretty close to declaring it is microtubules. And actually, Bruce is involved with this work as well.
Very good, very good. So plant cells have very few microtubules, but they do have them, particularly where the root and the stem join.
Thank you for a great talk and a wonderful conference. If I understand correctly, you take tubulin or tubules, microtubules, and send currents through them, electrical signals at different frequencies, looking for when the protein becomes conductive. Is that?
You're talking about Anirban's study. Sorry. Anirban's study. It puts a field, a voltage across, and then it becomes an alternating current, and at certain frequencies it becomes conductive. So those are the resonant frequencies.
So you're putting that stimulus in, the current in, but when these things are functioning in the brain, I assume there is some, like in a computer, you've got a clock that goes at certain gigahertz. What's the clock mechanism? Do these things, these gigahertz signals arise spontaneously, or what?
That's a great question, and Anirban should be the one to answer it, but he's developed this whole idea about time crystals, which have been around in physics for a long time, and he's shown that microtubules are time crystals, which is kind of the same thing, where you have clocks within clocks within clocks within clocks. And he's got all kinds of papers about this. So the microtubules are themselves time crystals. They are the clocks in the brain, and probably in biology in general.
Thank you.
Hi, Professor. Thank you for the talk. I was wondering if you can elaborate a little more on the study that's going to be conducted in India over summer.
Maybe Anirban could, but he's talking in the back of the room. They want to know about the study in India, putting patients to sleep with Okay, July will start. So it's going through ethical clearance in June, and we hope to start in July.
But what is it?
We're going to basically like this one is just going to be one probe, right? He's developing like a multi-probe. He's developing a multi-probe that can measure not only megahertz but gigahertz. But I think the first study will be just with a single megahertz probe on patients going under anesthesia in the operating room. Is that right? No. Oh, yeah, okay. He's talking about the fact, people say, well, that must be a muscle artifact, but it's not. And we have a video, I don't have it loaded up, of the same guy, Hide, holding it, and you see, and he's doing all kinds of blinking his eyes and twisting all around, and it stays exactly the same. So this should be, on top of everything else, better for brain-computer interface because there's no muscle artifact.
Okay, thank you. Looking forward to the results.
Thank you. All right. Thank you for a great session. Bruce for organizing and y'all for coming here.